Hello, good evening and welcome to our channel PW NEET English. I hope everybody of you are doing absolutely fantastic in your life and keep your focus and aim towards the preparation of NEET, and obviously together our effort can help us to crack NEET 2026 and the future years. So today in this session, this particular chapters will be covered by us, that is two chapters from class 12, that is human health and disease, and obviously the chapter called evolution. So Rudanjali, very good evening to you. Okay, so let's check who all are watching, from which place of India. Within 1 or 2 minutes, we will be starting the chapter Human Health and Disease. So in this chapter, we'll be talking about some common diseases in human being. What is the difference between disease, disorder, syndrome? What is the difference between symptom and sign? And obviously, with full focus, we will be covering two most important topics from this chapter. One is HIV/AIDS, another is cancer, and many more things. We'll be talking about alcohol and drug abuse and how to recover from them. Hello, Niharika, welcome again to this class. Vishnuvardhan, yes, welcoming you to this channel, and very good evening. Okay, so let's start the chapter Human Health and Disease. So, as we know, the definition of health already given by WHO, World Health Organization. So, when you talk about health, health means it's a state of complete physical, mental and social well-being. If you are only physically fit, and thinking that you are healthy, then it's not the proper criterion. Then you can call that person, if you are observing that person is physically fit only, but there is problem in social as well as mental well-being, then you can call that person as disease free. And already you have studied in some previous classes like class 9th or 8th what is the difference between healthy person and disease free person. Okay, so it is affected by a genetic disorder. When your daily easiness is affected by genetic disorder or infection caused by infectious agents or pathogens, as well as if you change your lifestyle, suppose if you are not drinking water properly, if you are not maintaining daily hygiene, if you are consuming alcohol and some narcotic drugs, then obviously they are gonna impact your overall health and cause you some disease. Okay, and if we want to link some prehistoric or some historic phenomena with modern day medicine, then it will be like this. We know that some Greek philosophers as well as ancient Indian Ayurveda gurus, they said that mind influences our immune system. So often you have heard this word that if you are taking too much tension, if you are in depression, or your life is going through a very hectic phase, then obviously you are gonna get weakness and then a disease. Why it's so? Because when we are in tension or depression, then our brain will be influencing our adrenal gland, and adrenal gland will be releasing a particular hormone called as cortisol. Yes, and the cortisol is called as stress hormone. Whenever you are in depression, tension or stress, your adrenal will be releasing cortisol, right? and your cortisol will be suppressing your immune system, and for a prolonged time, if you are in a stress, for a prolonged time your body is releasing, secreting cortisol hormone, and your immune systems or immune cells are suppressed, and it will be quite vulnerable. You will be quite vulnerable for the outer world pathogenic elements like bacteria, virus or protozoans to attack you, and that will cause you a disease inside. So that's why we need to differentiate between healthy person and a disease free person. Okay. Now, what do you mean by disease? When you talk this particular terminology, disease, it's a combination of two terms, dis and ase. Dis means just opposite, and ase means is the comfort of your daily life. So when those comfort of your daily lives are somehow disturbed by some pathogenic element, or by some nutritional deficiency, or with accompanying some internal organ failure. So that condition simply we call it disease. So as NCERT is saying that infectious or non-infectious diseases can be of two types, infectious disease and non-infectious disease. Okay, obviously we will be talking about all kinds of infectious and non-infectious disease in a brief mode, but can you tell me that what is difference between acute disease and chronic disease in comment section? Okay, physics and chemistry marathon classes will be there. Okay, I think the physics gonna start from the next week, and after that you'll be getting chemistry classes. Okay, so here I have a question. We know that there are two kinds of, you can, if you want to classify disease, you may get one group that is infectious disease or communicable disease, and another group that is non-infectious disease or non-communicable disease. Now, sometime you might have heard this particular terminology, acute disease and chronic disease. So, is there anybody who want to tell me the answer? What is acute disease and what is chronic disease? If you have studied class 9th biology, I think then it will be very easier for you to answer. I think all of you, like if you are watching this particular channel, then you are preparing for NEET, and obviously you have studied biology in class 9 in a very proper manner. So I think it will be very much easier for you to answer. Difference between acute disease and chronic disease. First tell me what is acute disease, and then tell me what is chronic disease. Niharika is saying acute disease can be cured. Exactly. Yes. If you are getting a disease very suddenly, then that disease can be called as acute disease, like common cold, like flu or influenza. These are some examples of acute disease. And obviously uh, chronic diseases not usually cured. Exactly, that's one of the very important fact about chronic disease. But the proper explanation for chronic, that means the chronic disease is developing inside your body for a long duration, and probably there is no proper cure. Yet we don't have any particular cure for that particular disease, that then that disease can be categorized as chronic disease. So acute disease will be appearing suddenly into your body, and within few days they will be, your body will be recovering from that disease. But there is no certain recovery for the chronic diseases like diabetes mellitus, like cancer, right? So these are categorized under chronic disease, or you can say emphysema. Emphysema is a disease which is caused, it's a chronic disease which is caused for prolonged smoking, right? You already studied in the chapter of breathing and exchange of gases. Okay. Now, if you look back into our history, then uh, the historic pioneers of medical biology, or medicine like Hippocrates and Indian Ayurveda gurus, they used to talk about good humor hypothesis. According to them, according to even Ayurveda, inside our body, there are three kinds of humor or fluids are there. Humor means fluid. So those three humors should be in a proper balanced mode in order to get a healthy condition. Okay, so that's called good humor hypothesis, and you need to remember the names. Who used to talk about good humor hypothesis? Greek philosopher and one of the pioneer of medicine, that is Hippocrates, and also Indian Ayurvedacharyas. Okay, so according to them, health is a state of body and mind, and that state of body should balance those three humors in a proper manner. Like one of those humors should be blood, another second kind of humor that should be your lymph kind of thing, they used to call it phlegm, and the third humor they used to call it black bile. Now this black bile terminology is used in our NCERT, and according to them, a person with black bile is depicting as hot personality, or the person is suffering from cough, cold and fever. Now you will say, sir, what do you mean by black bile? We know that there is bile juice produced inside our body. Bile juices are produced by our liver and stored into gallbladder. So is this black bile is similar to that bile juice which is blackish in color? Not really. Okay, so if you are vomiting, right, if you are vomiting or if you are defecating and there is bluish or blackish color into your vomit or into your stool, then they used to think that that is caused due to black bile formation into our body, and obviously that person is having hot personality. Okay, here hot personality means the person is suffering from cough, cold and fever. Yes. Now, later on, another scientist called William Harvey, we call him father of blood circulation, right? While he discovered the blood circulatory pathway, the double circulation of human being, William Harvey disapproved or disproved the hypothesis given by Hippocrates and Indian Ayurveda. So, good humor hypothesis was discarded later on by William Harvey. Okay. Now, by the time while William Harvey was doing his research, then thermometer was available, right? But when these Hippocrates and Indian Ayurveda gurus were talking about disease and health, then there was no invention of thermometer was there. So that was one of the advantage for William Harvey. By using thermometer, he took temperature of different person. Okay, even he observed that the person with black bile, means a person who is vomiting a blackish colored or deep greenish colored vomit, that person, the blood, the body temperature is similar, that is 37 degrees C. So he demonstrated that normal body temperature in person with black bile using the thermometer. Means if I am a normal person, then my body temperature will be 37 degree, and if you are a person with black bile, your body temperature will be the similar, that is 37 degrees Celsius only, right? So that's how William Harvey disproved that theory, that this presence of black bile inside the body is denotation of cough and cold or fever. Okay. So now we'll be talking about pathogens. So disease causing organisms. Disease causing organisms are called pathogens. So why they are called as pathogen? Again, we got this terminology from Greek word. Pathos means pain and gen means agent. So pain causing agent of your body. we call them pathogen. And it's written here, most parasites are pathogens. So see, parasites can be ectoparasites and endoparasites for us. Like if you talk about ectoparasite, bed bugs, mosquitoes, leeches, they are the examples of ectoparasite. And endoparasite, can you name some endoparasites? Can you write down in the comment section some examples or some names of endoparasites? You know about the disease malaria, right? So the malarial parasite is called Plasmodium, that will be attacking your liver cells as well as your RBCs. So as because they are staying inside your body, so Plasmodium is one of the very crucial example or important example for us, uh, that endoparasite, right? So Plasmodium is example of endoparasite. Yes, Ascaris lumbricoides, they will be staying inside your intestine mainly, right? If you are suffering from the disease ascariasis, so they are also example of endoparasites. So yes, if a parasite is staying inside your body mainly, so that is gonna harm your physiological system or the biochemical environment of your body physiology, then obviously you will be suffering from a disease. So that's why it's written here, most parasites are pathogens, means most parasites are disease causing agent. Like as I said, ascaris is a causative agent for ascariasis, entamoeba is a causative agent of one kind of dysentery, amoebic dysentery. Plasmodium is causative, is a parasite, endoparasite, is causative pathogen of the disease malaria. Okay. Now, if a pathogen is staying inside your body, pathogens have to adapt to live within the host. Like as Niharika has given the example, ascaris, right? It's staying inside our intestine. Now, intestine means a part of your alimentary canal, and your alimentary canal will be secreting HCl, highly acidic in nature, and other alkaline digestive enzymes and digestive juices. Now, if you are an ascaris, you have to protect your body from the attack of HCl, alkaline secretion, as well as digestive enzymes. So that's why we can observe that the outer coating or the outer covering of ascaris, that is made by thick cuticle, a waxy kind of layer, and they are preventing our digestive enzyme to act on or to digest the outer covering of ascaris. So that is the adaptation they have gathered over the time and due the course of evolution. Yes, Chaitanya, how are you? Very good evening. Welcome to the class. Okay. And if I talk about Tinea solium, Tinea saginata, another example of endoparasitic platyhelminths. Now, they're also staying inside our intestine, and your intestine will be performing peristalsis movement, and during the time of peristalsis movement, it will try to throw out the endoparasites out of your alimentary canal, and that's why Tinea has developed hooks and suckers for a grip or firm attachment with the inner wall of our alimentary canal or with the inner wall of our intestine. So this kind of adaptation or acclimatization can be observed for this endoparasite or for these pathogens. Okay. Now we can categorize this pathogen as bacteria, virus, fungi, protozoa, and helminths. Helminths means they are few ascalminths as well as platyhelminths. Like if I uh, name one of the example of bacteria, that is Vibrio, right? And Vibrio cholerae is a causative pathogen for the disease cholera, right? If we talk about virus, HIV virus, human immunodeficiency virus, that is causative agent for AIDS, right? Fungi, if I talk about trichophyton, epidermophyton, then they are causative agent of ringworm of skin. If I talk about protozoa, that malarial parasite plasmodium is one of the like endoparasitic protozoa, right? Helminth ascaris, causative helminth for the disease ascariasis. Okay, now in our NCERT we have to discuss about some common diseases in human being. So the first disease is given in NCERT that you can observe, that is typhoid. Now let's track the pathogen of typhoid. So the pathogen of typhoid is a bacteria, a gram-negative bacteria, called as Salmonella typhi, right? Okay, now let's check the mode of transmission, how a healthy human being can get that bacteria inside the body or get that infection, right? So it enters into the small intestine through the food or water and migrates to other organs via blood. So if you are asked that infection of typhoid is what kind of infection, then you have to answer it's waterborne or foodborne disease. Typhoid is a kind of waterborne or foodborne disease, right? Where it's, which organ is getting affected mainly? Mainly small intestine is getting affected, and that's why typhoid got another name, that is enteric fever. Enteric fever. Why we call typhoid as enteric fever? Because it's attacking the intestine. The other name of intestine is enteron. Okay. Now, if you are suffering from typhoid, what kind of symptoms can be observed? Now, you might have heard the terminology, another terminology which is kind of synonymous, that is called sign. So, what is symptom of a disease and what is sign of a disease? Can you comment down in the section of comment? What is the difference between a symptom of a disease and a sign of a disease? Or are they similar? Are they same? What do you think? Is there anybody who want to answer that difference between symptom and sign of a disease? In the meantime, we'll be discussing about the symptoms of typhoid. There will be sustained fever, sustained high fever of 39 degree to 40 degrees Celsius temperature, right? Sustained means prolonged fever will be there, accompanying headache, weakness and stomach pain, constipation and loss of appetite. And as because there is loss of appetite, you are not feeling hungry, you cannot eat, your digestive system unable to digest and absorb the food material, and that will cause weakness and stomach ache. Fever is a sign of disease. Fever can be sign, fever can be symptom, right? So difference between symptom and sign is the prospective. If I am the patient and you are the doctor, if I am the patient and you are the doctor, the kind of discomfort I am feeling right now, if I am a patient, then the feeling of mine will be called as symptoms, and as a doctor, it will be your scientific view to examine me, examine my discomfortness, right? So your vision about my discomfortness is called as sign of a disease, right? So it's a perspective what a patient is feeling, that is called symptom of a disease, and what a doctor is observing, that is called as the sign of that disease. Okay. Uh, So, let's check about few other symptoms observed during the time of this typhoid fever. Intestinal perforation and death may occur in case of severe infection. Intestinal perforation means if your intestine is too much attacked by the Salmonella typhi, then it will become porous in nature. There will be wound into your intestine, and that will call that perforation. And if intestine is perforated, that means your intestine is lost its capability to digest and absorb the food material, and that will cause your death. Okay. Now the confirmatory test of typhoid, you need to remember, that is called as Vidal test. Now why it's named as Vidal test? The process was told by a scientist, and his name was something Vidal. That's why we got the name of the confirmatory test for typhoid, that is Vidal test. And specifically what is done here, they will be taking your blood sample from your body and checking for the antibodies prepared by your body in order to combat the pathogenic proteins or pathogenic toxins produced by this Salmonella typhi. Okay. So if the laboratory people, the path lab peoples are able to find them, then it said that you are typhoid positive. If it's absent into your blood, then it said that you are typhoid negative. Yes. And one more important thing written in NCERT, there was a cook called Marie Mellon. Marie Mellon. She is also named as Typhoid Mary. Why so? Because Marie Mellon was a professional cook, but she was kind of unhygienic, very much unhygienic. She was a typhoid carrier who spread typhoid for several years through the food she prepared. Now, what made made her typhoid carrier? I'm talking about this cook called Mary Millon. Okay, maybe she was infected by typhoid long ago, but sometime what happens, a disease or pathogen will enter into your body, but it will it will keep quiet inside your body for you. It will not cause the symptoms, it will not cause the destruction into your physiology. So that's why you won't be feeling any diseased condition, or there will be no symptom found inside your body. Then in that case you will be said that you are a carrier of that disease, is because you are not suffering, but silently, hiddenly you are carrying the pathogens. Then the pathogen will make your body as a storehouse, as a reservoir. And whenever, if you are affected by Salmonella typhi, and if you are defecating and not washing your hand properly, and with that unwashed or improperly washed hand, if you are cooking food, then inside that food its possibilities are there that typhoid pathogen, that is Salmonella typhi, will gradually enter. And that's how she kept on spreading Salmonella typhi, and that's why later on when it's found, it's due to this similar cook into different families she was cooking basically, and then given the name Typhoid Mary. Next, another bacterial disease called as pneumonia. Pneumonia. Now check the name of the disease, pneumonia. This disease causing bacteria, generally they affects or they attacks our alveoli of lung. And you know that special cell that is present inside the alveoli, we call them pneumocytes. So the alveolar cells called pneumocytes are affected by the pathogen of this disease. That's why we have named the disease as pneumonia, and the pathogen is Streptococcus pneumoniae and another bacteria called Haemophilus influenzae. One bacteria Streptococcus pneumoniae, another bacteria Haemophilus influenzae. Okay, now it's a kind of airborne disease or droplet borne disease. It infects lung alveoli, and the alveoli get filled with fluid leading to respiratory problems. When your alveolar cells are affected, then obviously in the response of the inflammation, the pneumocyte cells of your alveoli will start releasing excess amount of fluid, and that accumulation of fluid inside the alveolar sac will be blocking your normal gaseous exchange. Okay. Now, it said that mode of transmission is by inhaling the droplets or aerosols released by an infected person. When a person is sneezing or coughing, okay, if I am a person who is infected by pneumonia, and if you are staying inside my room, then while I am coughing or sneezing, the droplets gonna spread through the air, and if you are inhaling the similar air, obviously Streptococcus pneumoniae gonna enter into your lung, your alveoli, and again it will cause pneumonia inside your body. Okay, or maybe possibilities are there, sharing glasses and utensils with an infected person. So the disease can be airborne, the disease can be droplet borne, the disease can be fomite borne. What do you mean by fomite? The utensils, or the panes or the glasses used by the patient, they are called as fomite. So next thing, what can be the symptoms for pneumonia? Now, if you are going out of oxygen because your alveolar surface is blocked already by the alveolar fluid secreted in response to the inflammation or infection of Haemophilus influenzae or Streptococcus pneumoniae, then there will be respiratory problems. So there will be respiratory problems, fever will be there, chills, you are feeling cold, cough will be there, headache will be there, and in the severe case, lips and fingers and nails turn grey or bluish. Why the lips and fingers are turning gradually greyish or bluish in color? As because your tissue inside your body are not getting supply of adequate oxygen, and that's causing hypoxia. Into your body, and the symptom of hypoxia is blue colored lips, blue colored nails, even eyes, right? So that was all about this disease pneumonia. Now we'll talk about some other bacterial diseases. First, mentioned in NCERT, that is dysentery. Okay, now there are two kinds of dysenteries we need to talk about. One is bacterial dysentery and another is protozoa cause dysentery. Okay, now for this dysentery caused by bacteria called Shigella, or a specific virulent strain of Escherichia coli and few strains of Salmonella, and you may get this infection by contaminated water and food. This dysentery is caused by bacteria, Shigella, Escherichia coli, Salmonella, or all of them are different virulent or the infectious strains of bacteria. Now, this, when you're talking about this E. coli bacteria, normally it's quite good bacteria. It's a symbiont for your gut, right? It's a symbiotic bacteria inside our alimentary canal, but uh, some modified, some mutated E. coli bacteria can cause dysentery. Now, what are the symptoms of dysentery? We call it loose motion, excessive loss of body fluid and electrolyte with defecation or while you are passing your stool or fecal matter. Okay, there will be infection into your large intestine, Another bacterial disease quite dangerous, that is plague. Okay. Now plague is caused by Yersinia pestis, and the vector for Yersinia pestis, the bacteria, will be carried and will be carried by vector called rat flea or xenophila. Okay, so what is vector? This rat flea, generally they will be harboring into the skin of rats or mouse. Now, if the rat is infected by Yersinia pestis from rat while it's sucking the blood of rat, this Yersinia pestis, this rat flea or xenophila will take the Yersinia pestis into its body, and again when it's gonna bite another healthy human being, during that time it will be infecting or it will be injecting the Yersinia into the blood of that healthy human being. And this rat flea or xenophila, this particular organism, this particular fly is also being affected by Yersinia pestis. So that's why we call it as vector. Okay, so there is a difference between carrier and vector. Carrier organism means like normal house flies. They are carrying germs to you, but they are not getting, those house flies are not getting affected or infected by the disease causing pathogen. Then simply we will be calling them as carrier of a disease. Now what is the difference between carrier and a vector? A vector is disease fetching or the pathogen fetching organism to you, and that particular organism is also infected by that pathogen. Like if you are getting suppose typhoid, or if you are getting entamoeba histolytica, which is a causative pathogen for amoeboic dysentery. Now that is fetched to your body with by that house fly, right? But the house fly is not infected by that particular pathogen called entamoeba histolytica. So in that case we say that the house fly is acting as only the carrier of the disease. But here in this case it's written as vector. That means the rat flea is fetching Yersinia pestis to us, and this Yersinia pestis will be infecting human being as well as this rat flea. Another bacterial disease that is given in our NCERT, that is diphtheria, and the disease causing agent or the disease causing bacteria is Corynebacterium diphtheri, and this Corynebacterium will be harboring into your tonsils, right, into your lymphoid organs here near to your throat and your tongue. Next, now the next disease is given in our NCERT, that is common cold. Now common cold is not a bacterial disease, it's a viral disease caused by rhinovirus. Now why the virus is called as rhinovirus? Rhinovirus is a special group of virus which are modified to infect our nose region. That's why we call them rhinovirus. Okay, so they infect nose and respiratory passage but not lungs. So that's why they got this name. They will be infecting our nasal passage and nose only, but they will be not going towards your alveoli and bronchi and bronchioles. Okay. The mode of transmission is simply inhaling droplets resulting from cough or sneeze from other person through contaminated objects like pens, books, cups, or doorknobs, computer accessories, and simply we also call these things as fomites. So like pneumonia, common cold is also droplet borne disease as well as fomite borne disease. And symptoms for this particular disease gonna be simple nasal congestion, nasal discharge, congestion and discharge of mucus, fever, headache, sore throat, cough, hoarseness, tiredness, and common cold last for 3 to 7 days. And if you are taking antibiotic, if you are not taking antibiotic, doesn't matter. Within 3 to 7 days, automatically your body is gonna recover that. Okay. Now, I have a question here. So, if you are knowing that this common cold is caused by a virus called rhinovirus, but still when you are visiting a doctor will be prescribing you some antibiotics, azithral, azithromycin, okay. And we know that those antibiotics are not effective against viruses, but antibiotics can kill bacterial infections, right? But after that also, why doctor prescribing us antibiotics even if you are affected simply with rhinoviruses or simply common cold? Can you give the answer? For viral infection also, for season, during the time of season change, for viral infection, when you visit a doctor, they just prescribes us azithromycin, right, or azithroclavulin, some sort of antibiotics. And already I think many of us, we know that antibiotics are effective or can kill bacteria, but antibiotics cannot kill virus. So still, why doctors or medical practitioners are prescribing us? Can you tell me the answer? Okay, Prabhakaran is saying because virus mutate fastly, so. But you know that the antibiotics you are taking azithromycin, generally nowadays doctors are giving either azithromycin or clavulanic acid as antibiotic, the tablets you are eating, right, apart from the paracetamol and all. Mm, the rhinovirus are 69 type there. Okay. No, Prabhakaran, that's not the phenomena, because these simple antibiotics cannot affect virus, cannot kill virus, right? So there is no meaning of taking antibiotic for just for viral infection. Okay, yes, Niharika got the right answer, so that it does not cause any bacterial disease like pneumonia. Yes, whenever there is simple, normal virus infection or viral fever, then anyhow your immunity is somehow weak, right? So that's why, as a precaution, doctor is prescribing you those antibacterial agents called antibiotics, so that further you won't be getting any sort of bacterial infection, like you will not be infected by salmonella, infected by Haemophilus influenzae kind of bacteria, or causative diseases like pneumonia. That is the correct answer. Okay. Now we will be talking about another disease called dengue. Okay. So these two diseases are newly incorporated. One is dengue and another is chikungunya. Now dengue and chikungunya both are vector-borne disease. Vector-borne disease means there is some other animal acting as a vector to fetch the disease-causing pathogen to you. And the vector here, that is mosquito. So vector is Aedes aegypti mosquito, a special species. Okay, then the dengue is caused by a special virus called as Den virus, belong to the group flavivirus. And the symptoms, if you are suffering from dengue, there will be hemorrhagic fever. Hemorrhagic fever means there will be internal blood clotting should be there. Severe headache will be there, muscle and joint pains due to the toxin produced by these flavivirus, and rash upon skin, and retroorbital pain, and plasma leakage should be there. Okay. Now if we talk about chikungunya, chikungunya is caused by a virus called alpha virus. Though chikungunya is also fetched to your body, the chikungunya causing virus is fetched to your body by Aedes aegypti mosquito, and the symptoms are severe joint pain, high fever, rash, muscle pain and headache. Okay, so that was about dengue and chikungunya. Now we'll talk about very important disease as per NCERT, and it's a protozoan disease. Okay, and that is malaria. Now, pathogen for malaria is Plasmodium. Plasmodium is a protozoa. Plasmodium is a protozoa, not a bacteria, not a virus. And the common species of Plasmodiums, we need to talk about Plasmodium vivax, Plasmodium malariae and Plasmodium falciparum as given in NCERT. Okay. Now, out of this vivax, malariae and falciparum, which is the most dangerous strain of Plasmodium, that is Plasmodium falciparum. Yes, that can cause malignant, means very dangerous kind of malaria, and that can cause even death. Okay, so that's why it is considered that this Plasmodium falciparum is the most dangerous strain of Plasmodium parasite. Okay, so it's written here, most serious or malignant malaria is caused by Plasmodium falciparum. Okay. Now, when you are getting the disease malaria, generally the disease will reappear into your body within 3 days or 48 to 72 hours. Okay. Within every 3 days or 48 to 72 hours, there will be relapse of fever. There will be recurring fever, right? Again and again you are getting the fever. So that is the crucial symptom of malarial disease. And the phases of that fever will be like this. First, there will be chill phase or cold phase. First, there will be cold phase. You will be shivering, right? And second phase of the fever is called hot phase. And in this hot phase, there is a sustained fever of high temperature over 102 degrees Celsius at least for 4 to 10 hours. And suddenly, after 4 to 10 hours, you will feel somehow relaxed and there will be sweating. So there will be a sweating phase. Okay. And within every 3 days or 48 to 72 hours, this kind of symptoms will be repeated. Okay. And you know that mode of transmission is by female Anopheles mosquitoes, as because why not male Anopheles mosquitoes are vectors for this disease. Chikungunya and dengue is not given in NCERT, but for if you follow the NTA syllabus for our NEET, then you have to study those two diseases, chikungunya and dengue, right, Niharika? Okay. Now here I got a question for you. Why the the vector for malaria is female Anopheles mosquito? Why it's not male Anopheles mosquito? Why always female mosquitoes? Why always female mosquitoes causing diseases? Can you answer? Because male cannot drink blood. Yes, exactly, Prabhakaran. Because males are generally not fond of drinking blood. They are happy with fruit juice. Males, they are happy with juices from plants, right? But for the maturation of the ovary, yeah, male is vegetarian. And why females are sanguivore? Why female mosquitoes are blood sucking in nature? Because they need the warmth of mammalian blood and calcium and few other minerals present into our body, and that will be helping the female mosquitoes to mature their ovaries, and which is necessary for the maturation of the eggs. That's why they are bound to drink mammalian blood. Yes. Okay. And one more thing you might have observed, that if you are standing alone near to a field and suddenly some mosquitoes will be hovering around you. Okay, so how they got to know the presence of human being or presence of mammal? Have you ever wondered? Have you ever questioned? Like uh, there is a room, and you thought I'll be sleeping for some time, right? And suddenly you found that mosquitoes are hovering around you, singing and dancing and biting and making a chaotic condition. Okay, so how they got to know the presence of human being or presence of you? And they are flying very rapidly, flying or hovering nearby you. It's your odor. Okay, odor can be one thing, but mainly they are attracted by, mainly mosquitoes are attracted by the carbon dioxide we release during the time of exhalation. They can sense that gradient. Okay, now let's talk about the symptoms of malaria fever. Okay, so hemozoin, a toxin released by plasmodium, causes chill and high fever recurring in every 3 to 4 days. Okay, that's why we call the malaria as tertian malaria. The malaria caused by these plasmodium vivax, that is called as tertian malaria. That means in every 3 days or every 3 to 4 days there will be recurring fever. Remember the malaria, the specific kind of malaria which is coming again and again in every 3 days, that is called as tertian malaria. And how these hemozoin toxins are formed into our body? When this plasmodium will be attacking your RBC, they will be feeding upon your hemoglobin molecules inside the RBC. And like you, they are also digesting, they are also excreting. So basically, these hemozoin molecules are their excreta, and those excreta of plasmodium is harmful for you. When they are releasing the hemozoin into your blood, your hypothalamus can sense the change, and that will be causing a change of your body temperature. Okay. Now, if we talk about the life cycle of plasmodium, life cycle of plasmodium, plasmodium vivax. Okay. So this is your hand, and that female Anopheles mosquito is biting you. And that female Anopheles mosquito is biting you. Now, while this female Anopheles mosquito is biting you, it will throw some saliva of it inside your bloodstream just prior to sucking the blood from you. And the salivary glands, remember, the salivary gland of female Anopheles mosquito, the infected Anopheles mosquito is containing a special stage of Plasmodium called as sporozoite. And these sporozoite stages are inserted into your bloodstream. So the infectious stage of a Plasmodium, that is the sporozoite. You need to remember sporozoite, the infectious stage of a Plasmodium. Okay. Now, as soon as this sporozoite enters into your body, your macrophage, your immune cells will try to catch and kill sporozoite. So, to survive, these sporozoites will run and will search for a safe place where your body's immune system not gonna disturb. So, they will be finding a better place where they can increase their population, and that better place is liver. Now, why they have chosen liver initially after entering into your body? Because the cells of liver, we call them hepatocyte cell. The cells of liver, we call it hepatocyte cell, rich in a special kind of nutrient called glycogen, right? Okay, the polymer of glucose. So this glycogen are quite nutritive for this sporozoite stage of Plasmodium. Now these sporozoites will be entering into the liver cells. They will be feeding upon the glycogen, and gradually they will increase their number by the process of multiple fission. You know that amoeba can perform binary fission, multiple fission. So this Plasmodium, the sporozoite, they can reproduce asexually, and the mode of asexual reproduction here, that is kind of multiple fission, and we named that multiple fission as schizogony. Okay, so what they will do, they will perform schizogony, and performing schizogony, they will become another, they will change their shape, they will change their morphological structure into the next phase, and we call them as merozoites. Okay. Now few of these merozoites will go back to the liver and attack again, and few of these merozoites, they will be attacking our RBC. Few of the merozoites, they will be attacking our red blood corpuscles. Okay. Now while these merozoites are attacking red blood corpuscles, they are feeding upon the hemoglobin present inside, right? And when the hemoglobin is finished, they will rupture RBC. When the hemoglobin is finished, they will break down RBC, and in that meantime, some toxin is released into your blood, that is called as hemozoin. Toxin, and this hemozoin toxin is responsible for fever into your body. Okay, now what's happening with this Plasmodium present into the RBC? Okay, now few of the merozoites will come out from the RBC and will attack more RBC, attack some other RBC. Few of the merozoites will go inside the liver and will feed upon the glycogen present into the hepatocytic cell, and few of the merozoites few of the stages of Plasmodium present inside the RBC. Actually, we call the stages as rosette stage, signet ring stage, because while these plasmodiums are entering into the red blood corpuscle, it will look like a ring. It will make a ring like structure inside RBC, and it's very much hungry in nature. That's why we call it trophozoite stage. Now inside RBC it's not merozoite, the stages of same plasmodium we call it trophozoite. Why it's trophozoite? Because it's very much hungry, it's always eating upon the hemoglobin molecule. And the next stage that we will get, they are precursor cells of ovum or sperm making cells of plasmodium. Okay, so those cells are called as megagametocyte stage of plasmodium and microgametocyte stage of plasmodium. Gametocyte stage of plasmodium. So inside our RBC, these two very crucial stages will be formed, megagametocyte and microgametocyte. Then after RBC, they will be spread with your circulatory system, with your circulatory fluid. Okay, so that was all about the cycle of plasmodium inside human body. Again, I am repeating it. How it's entering into your body? It's entering through a female Anopheles mosquito bite. Okay, and the stage is called as sporozoite stage. The stage is called as sporozoite stage. Now, entering into your bloodstream, that will be chased by your immune cells, your phagocytic cells. So these sporozoites want to survive, want to escape now. Now they are searching for a place where these immune cells will not be attacking them into human body, and they can get a good amount of food. So they are finding liver as their savior. So they will be entering into the liver cells, and then inside the liver they are getting these glycogen molecules as their food, and by feeding them they will increase their population. Now they will destroy the liver cell and come out as another stage that is called as merozoite. So after sporozoite the stage is called as merozoites. Now few merozoites will affect some other liver cell, and few merozoites will enter into your bloodstream to attack your RBC, red blood corpuscles. Okay. Now while these merozoites are attacking your RBC, then no more we will be calling them merozoites, we will be calling them trophozoite, because they are feeding on the hemoglobin of RBC. Now they are feeding hemoglobin and they are destroying RBC, and they are releasing their excreta as a toxin, and this toxin is called as hemozoin toxin. And this hemozoin toxin is responsible for the fever, and that's why we are getting this malaria disease, right? Okay, now further stages of trophozoite, they will multiply inside the RBC. Few will get back to the liver, few will attack some other RBC, and few will become a large cell called as megagametocyte. Few will become a large cell called megagametocyte, a few will become a small cell called as microgametocyte. Now basically this megagametocyte and microgametocyte, they are special stage of plasmodium which can produce ovum and sperm, but not inside human body, when they will get inside the body of mosquito. Now how they will be going inside the body of mosquito? So from here a new stage will start in their life cycle. Now suppose this particular malaria infected person was bitten by another healthy Anopheles female mosquito. So while this healthy Anopheles mosquito is biting that malaria infected person, while she is sucking blood, she will be getting the megagametocyte and microgametocyte stage into her body, right? Now, getting inside the body of mosquito, this megagametocyte and microgametocyte will enter into the gut, means will enter into the alimentary canal of that female Anopheles mosquito, and the environment of gut will promote formation of ovum and sperms. Okay, from which cells? From megagametocyte there will be formation of ovum, from microgametocyte there will be formation of sperms. And as you know that after the fertilization there will be formation of zygote, and this zygote gradually will modify one enlarged structure called eukinetes. This eukinetes will be modified into oocyst. Okay. So basically these, like for human embryo we have different stages. After zygote we have morula, then we have blastocyst. Similarly, the different phases of the embryo of this plasmodium upon fertilization of ovum and sperm will be zygote, then eukinetes, then oocyst. Okay. Now from the oocyst there will be formation of thousands of sporozoites. Inside the oocyst there will be formation of thousands of sporozoites. Okay, by spore formation is a kind of sporulation process, and these sporozoites will enter into the salivary gland of that mosquito. And when this mosquito is biting another human being, it will be delivering the same sporozoites present into her salivary gland into the bloodstream of that particular human being. So that's how the life cycle of plasmodium continues into the body of human being as well as continues into the body of mosquito. Okay. So if I summarize this, the life cycle of plasmodium into the body of human being and into the body of mosquito, it will be like this. Here in this side we will be talking about plasmodium life cycle inside human being. Okay, so inside human it will be like this. The stage which is entering, that is called as sporozoites. Okay. Now these sporozoites are entering into the liver. Okay. Now from liver, the stages of sporozoites they are coming out, we call them merozoites. Okay. Now these merozoites will attack RBC, red blood corpuscles, right? Now from red blood corpuscles, the stage which are coming out, we call them gametocytes. Okay, gametocytes are of two types. Gametocytes are of two types, megagametocyte and microgametocytes. Okay. Now what will happen? This megagametocyte and microgametocyte, through a mosquito bite from the human body, it will enter into the body of mosquito. Now entering into the mosquito alimentary canal, into the mosquito gut, this megagametocyte and microgametocyte will form ovum and sperm. Upon fertilization, there will be formation of zygote. This zygote will be developed into a structure called as eukinetes. This eukinetes will be modified into oocyst inside the mosquito gut or mosquito gut wall. Now this inside this oocyst, a special structure of the embryo of Plasmodium, there will be formation of sporozoites. Now, where inside the mosquito body these sporozoites gonna escape or uh, gonna make their harbor into the salivary gland, and when the mosquito is biting a human being from the salivary gland, these sporozoites will enter into another human being again. Okay, so these are the stages of what? These are the stages of Plasmodium. These are the stages of Plasmodium, life cycle of Plasmodium. So one part taking place inside the human being, and if you are asked which phase of the life cycle is taking place inside the human being, there is the asexual mode of their reproduction, asexual phase of their reproduction taking place inside the body of human being. Now, sexual phase of Plasmodium is running inside the gut and body wall of mosquito. So, we can observe that for this particular pathogen Plasmodium, this particular endoparasite is having two hosts. One host is human being, another host is mosquito. So this kind of parasites can be called as so this kind of configuration, this kind of having two hosts called as digenetic host. Digenetic means Plasmodium is a single parasite. It's having one host as human, another host as mosquito. Mosquito is acting as a primary host and human is acting as a secondary host. Means inside the body of mosquito, it's performing, Plasmodium is performing sexual mode of reproduction. Inside the body, inside the secondary host, it's performing asexual mode of reproduction. Okay. Now I'll talk about amoebiasis. Okay, so the pathogen for amoebiasis is another protozoa called as Entamoeba histolytica. See the name. It's a kind of large amoeboid protozoa, and it will be affecting your gut or alimentary canal. In Greek we call it enteron, that's why it's called as entamoeba. An amoeba which is attacking your enteron or gut. Why it's called as histolytica? We know histology is all about study of tissue. Now while it's attacking you, it will be disturbing or destroying the tissue of your gut or alimentary canal. That's why it's called as histolytica. Lysis means destruction, right? And histo means tissue. Now let's talk about the mode of transmission, how will be infected. Okay, so we got mechanical carrier, and they are house flies. House flies transmit parasites from feces to food and water. So these are carrier borne disease, as well as you may get directly by eating contaminated water as well as food. Okay, now if you are getting amoebiasis or amoebic dysentery, amoebiasis is also got another name, amoebic dysentery. Dysentery, you know that excess loss of water or liquid fecal matter. Okay, so symptoms are firstly constipation, then there will be abdominal pain, your cramp inside your intestine, right? Stool with excess mucus and blood clots. Now why excess mucus and blood is coming through your stool? As because this Entamoeba histolytica is causing destruction of the wall or the mucosal layer and submucosal layer of your large intestine. Next, ascariasis. Now ascariasis, helminth borne disease. Okay, pathogen is ascaris. Helminth disease means ascaris the common roundworm. The scientific name of common roundworm is Ascaris. So they are intestinal parasite and they belong to the phylum Ascalmins. Okay. Now how you may get this Ascaris? From soil, water, vegetables, fruits, etc., or through contaminated, when it's contaminated by fishes containing egg of parasite. Now how these food, water or vegetables are contaminated by fecal matter or feces, right? If somebody is having ascaris into his or her intestine, and the disease is called ascariasis. If someone is suffering from the disease ascariasis, now he or she went for defecation into a vegetable field, into open field. Now you can guess that what is gonna happen. Now he or she will be defecating or the contaminating of the vegetable, the leaves of the vegetables, of the veggies or crops. Now if you are collecting the vegetable after some days and without washing it properly, without boiling it properly, if you are consuming that, you may engulf some eggs of ascaris, and upon going inside your intestine, the eggs will be hatched and the larval phase of ascaris will attack your intestine, and the symptoms are internal bleeding, muscular pain, fever, anemia and blockage of intestinal passage. So it will enter through your intestinal wall, it will be attacking your liver, it will be attacking your spleen, sometime it can reach into your eyes as well in severe condition. Okay, and sometime it will be reaching into your muscles as well, that will cause muscular pain, and fever is very common symptom for all kind of infection. Why it's so? Fever is a very common symptom for all kind of infection because when a pathogen is entering into your body, it will release the toxin, right, which we call antigen. Okay, now your hypothalamus is continuously monitoring your blood quality. Now if there is something bad present into your blood, then your hypothalamus understands, I need to increase my body temperature so that that increased temperature may kill the pathogen which is causing destruction inside the body. So actually your hypothalamus, or that is called also called as thermostat of our body, is causing fever for good for us. Fever is good for us, right? It's not a disease. Fever is symptom, and that is the mechanism of your body. By increasing the temperature, your body wants to kill the pathogen. Next, we will talk about filariasis. Okay, filariasis again caused by another roundworm called Wuchereria. So the pathogen for filariasis is filarial worm called Wuchereria, and the common species are Wuchereria bancrofti and Wuchereria Malai. Okay. Now mode of the transmission of this Wuchereria is from the bite of female Culex mosquito. Again, this female Culex mosquito, they are acting as the vector for this particular disease, and symptoms are like this. Filarial worms live into our lymphatic vessels, usually towards the lower limbs or lower parts of your body, because upper parts of our body during the time of breathing movement, during the time of walking, running, or while you are eating, there will be too much disturbance in upper part. But for our lower part, while you are sleeping, there will be less movement happening. So that's why they prefer to stay inside the lower limbs or inside the genital organs like inside penis or scrotum. Okay. So chronic inflammation of these organs in which they live for many years, so limbs and genital organs may be deformed for a long time if these filarial worms are harboring inside your limbs. Then they will cause inflammation and gradually swelling of your legs, swelling of your limbs, and that's why we call the disease as elephant feet disease, or also can be called as elephantiasis, due to the swelled uh, uh, hind limbs, right? Okay. Now, another disease called as ringworms, though it's named as ringworms, but they are not caused by worms, rather they are caused by fungi. Yes, ringworms are caused by fungi. So pathogens are given, name is given here: microsporum, trichophyton and epidermophyton. Remember these three species of fungi: microsporum, trichophyton and epidermophyton. These are responsible for this ringworm. Okay, now where you may get the infection? They are seen in groin, in between toes. Now, the safe places of your body, the hidden places of your body, where gradually they will be getting more warmth and moisture, right? So that's why from soil or by using towels, cloths or comb of the infected person, you may get this infection. Heat and moisture help fungi to grow, and heat and moisture is very much important for this for the sporulation and for their establishment of their colony. That's the reason they will be choosing this kind of part of your body. Okay. Now symptoms when you are affected: there will be dry scaly lesions on skin because the epidermis is already damaged by them, nails, scalp, etc., and with intense itching will be there because inflammation is going on. Okay. Now here I got a question: which of the following disease is a non-infectious as well as the major cause of death in humans? So can you write down the answer into the comment section? Okay, so I got the answer from Niharika, Prabhakaran and Jasmine. Okay, so you all have given correct answer, that is cancer. Okay, now you got another question. Assertion is saying that balanced diet, personal hygiene and regular exercise are important for maintaining good health. And the reason is saying that health can be maintained only through vaccination and awareness about disease. Now please, I think it's a easier one. You can answer easily. Please try to give the answer into the comment section. Okay. So Niharika has given the answer as option 3 and Prabhakaran is saying is option 2. Okay. So see, balanced diet, personal hygiene and regular exercise are important for maintaining good health. This is true. Health can be maintained only through vaccination and awareness about diseases. That is false. Okay. So assertion is true, but reason is false is the correct answer. Yes. Now, next question, Entamoeba histolytica. Entamoeba histolytica, right now we were talking about the disease. The pathogen of the disease is a parasite of large intestine, liver, lungs and kidney. Can you answer? Okay, got it, Prabhakaran. Now, can you answer this question? Entamoeba histolytica is parasite of, the name itself is saying the answer. Entamoeba, Entamoeba. If you split the name, you will get the answer. Yes, Niharika has given the correct answer and it's large intestine. Okay. Now we'll talk about immunity. So every day we are exposed to large number of infectious agent. However, only a few of this exposure result in disease. Now this is a question asked by NCERT. So thousands of disease causing pathogens are roaming here and there. Okay, and they are trying to penetrate inside your body and try to cause disease, and sometimes they are trying to kill you, but you are absolutely safe. But what is the reason? Because we have immunity, we have immune system present inside our body. We have immune system present inside our body. Okay. So if we talk about our immune system or immunity, it is giving us the capability to fight against infection and fight against diseases. That capability is called immunity, and the system is conferring us to give that immunity, that is called as immune system. Okay. So when you talk about immune system or immunity, there are two kind, innate immunity and adaptive immunity. Innate immunity and adaptive immunity. Okay. Innate immunity means the specific kind of immunity you are getting from birth. The immunity already present into your body from your time of your birth, that is pre-installed into your body, right, from your birthday. And adaptive immunity is quite different from innate immunity. Adaptive means after the birth, if the child is gradually acclimatizing and fighting with the infection, right, first we are getting infection and then our body is managing to produce some specialized cells to cope or to fight those diseases or to fight those pathogens, and that immunity is called as adaptive immunity, or you can also call this as acquired immunity. So adaptive immunity is acquired in nature and innate immunity is inborn in nature. Now let's study in detail about the innate immunity and acquired immunity. So innate immunity is written as non-specific immunity present at the time of birth. Okay. Now what do you mean by non-specific immunity? Uh, for the cells of adaptive immunity, the prime cells of adaptive immunities are T cell and B cell, and there will be specific T cell or specific B cell or specific antibodies produced against some specific disease. For adaptive immunity, our body will be producing antibodies, right, to recover us from diseases. And how these antibodies are made? Antibodies are made upon the interaction with disease causing organism. Like if you are infected by typhoid, your adaptive immunity will be making typhoid specific antibody. If you are infected by malaria, your body will be making the plasmodium, the toxin produced by plasmodium, that plasmodium specific antibodies inside us. But if we talk about the components of innate immunity like macrophage, like neutrophils, the phagocytic cells of your body, some WBCs, like you know about macrophage, you know about neutrophils. So they cannot differentiate between a bacteria of typhoid or a bacteria of pneumonia. So they can kill, they will try to kill all kind of pathogenic bacteria or all kind of pathogen which is trying to enter or which is already entered into your body. So that's called the non-specific immunity. They cannot recognize a specific bacteria and they cannot differentiate a bacteria from other. That's why it's written here they are non-specific immunity. And for innate immunity, we have to talk about 4 types of barrier. Means if you are a bacterium and trying to enter into a human body, you have to cross 4 types of barriers. So what are they? The first barrier you need to cross, if you are a bacteria, this is the bacteria and it's trying to enter into your body, then the first barrier you will be facing, that is called as physical barrier. What are the function of these physical barriers? They prevent entry of microbes like through skin, mucus coating of the respiratory tract or gastrointestinal or urogenital tract. This mucus will be trapping microbes. So our skin and mucosa layer of this 3 system, mucosa present into our respiratory system, mucosa present into our digestive system or inside gastrointestinal tract, mucosa layer present into our reproductive system lining, that is urinogenital tract. So these 3 are the passage of entry of many pathogens. So whenever a pathogen will try to enter through your respiratory system or alimentary canal or your reproductive tract, then your mucosa layer will secrete lot of mucus and block the entry. And that is the reason whenever you are falling sick, suppose you are you got the disease common cold and disease, you got the disease pneumonia, there will be formation of a lot of mucus into your respiratory passage, right? Okay, simply even your skin is maintaining a rough surface, an acidic surface. Your skin, surface of your skin is quite acidic in nature, right? So the skin will try to block the attack of pathogens. So these 2 are our prime physiological barriers as per NCERT. Now if you are, if as a bacteria you have successfully penetrated this physical barrier, then a second barrier will try to stop you. Then that barrier should be physiological barriers. Now what are they? So physiological barriers, they prevent microbial growth inside our body, like gastric HCl, the hydrochloric acid secreted by your stomach, or the saliva or the tear, they are called as physiological barrier. Even fever is also example of physiological barrier. Even fever is also example of important physiological barrier. See, as I said that fever means your body will increase the temperature and it's trying to kill the pathogen inside, right? With food, if you have ingested some microbes, some pathogens, then HCl will try to kill that. Your saliva will try to kill that, because saliva and your tear is containing a special antimicrobial enzyme called as lysozyme, and that lysozyme present into your saliva, lysozyme present into your tear is continuously helping you to kill bacterial attack. Okay, now that pathogen has penetrated the physical barrier as well. Now there will be a third barrier, they will try to stop the attack of the pathogen or the bacteria. So those third barrier is called as cellular barrier, some specialized cell. And let's talk about those specialized cell. They are mainly phagocytic cells, like few of the WBC. And who are phagocytic WBC? You know, they are neutrophil cells. They are monocyte cell, neutrophil monocytes. Monocytes can be also called as macrophages. They can move like amoeba. They can perform phagocytosis with the help of their pseudopodia, and they can engulf the pathogen. And also some natural killer cells or NK cells act as your cellular barrier. Now if you don't understand what phagocytosis is, if this is the macrophage, this is how your macrophage looks like, and this is the bacteria, then gradually what will happen, your macrophage gonna engulf the bacteria and kill it. Your macrophage will engulf the bacteria and will kill it. So that was about cellular barrier. So remember important cells of your innate immunity: the prime phagocyte, that is neutrophil, then monocyte or macrophage, and do not forget the name of NK cells, natural killer cells. Okay. Now we have a fourth kind of innate immunity barrier also present into our body, and what is that? Okay, so that barrier is called cytokine barrier. Now you have to understand what do we mean by cytokine. Cytokine are some special chemicals released by some special cells. See how our body will be producing cytokines and when our body will be producing cytokines. So virus infected cells secretes a cytokine protein called interferons. So interferon is example of cytokine. Which cells are producing cytokines? Virus infected cells are producing cytokines. Why it's so? Because it protects non-infected cells from further viral infection. These cytokines will be promoting, will be telling the neighboring cells, please activate your immunity protocol so that you will not be getting infected by that same virus. Okay. Now we will talk about acquired immunity or adaptive immunity. Now important thing about acquired immunity is pathogen specific immunity developed during our lifetime. Now what do you mean by pathogen specificity? Our body will develop this particular kind of immunity, and this is different for different kind of pathogen, right? That's why it's pathogen specific. So it's characterized by memory. So during first encounter of a pathogen, your body produces primary response in low intensity, but the second encounter of the same pathogen causes a secondary or anamnestic response in higher intensity. So the graph of your acquired immunity will be like this. Suppose A is the point where your body got affected by a particular virus. Suppose it's a chicken pox virus. Your body got affected by chicken pox virus. Now against that chickenpox virus, your body will start producing some. Your body will start producing some what? Some antibodies. Now this is the peak point. Your body will produce uh, much number of antibodies to fight against the chickenpox virus, and as a result gradually you are getting rid of the disease. Your disease is recovered now. Okay, so this is called the primary response. This is called the primary response of the disease. What is response of your immunity? When you are falling ill, your body against that will start producing antibodies to fight the pathogen. So that is called the primary response. Okay. Now once you are experienced that this kind of pathogen is there and that can attack me, so you will prepare yourself in a better manner, so the next time you won't submit your body immune system won't be submitting your physiological condition to the disturbing or to the disease causing pathogen. Similarly, suppose you are attending an exam for the first time and you are unaware about the type of question that may come. Okay, so for the first attempt of your exam, you were not able to score good marks. Okay, now once you are experienced, is this kind of question may be asked. So after that, you will start preparation for second attempt, and in the second attempt, without any doubt, you scored a good mark and you have secured a good rank as well. So the first exam and your effort was first response or primary response. The second exam and against that your good score was your secondary response. So similarly, what happens with our acquired immunity or adaptive immunity? Now at the point B, at this point, again the same virus is attacking you, the same chickenpox virus is attacking you. But this time what your body will do, very quickly your body will produce very high amount of antibody in this point. Okay, and very quickly you will be recovered from that particular disease, or you won't be getting any disease at all. So this is the secondary response. This is the secondary response, or also called as anamnestic response. And an amnestic response means a response which is having memory. Okay, first your body was submitted in front of the pathogen, but once it's recovering you for the second time, your body is already ready or equipped to combat that disease. So for the second time when the disease is trying to attack you, what your body did, your body produced too much amount of antibodies, and we call that anamnestic response, and those antibodies will be rapidly will be eradicating or killing the second attack of the same pathogen. Okay, so that is all about the memory characteristics feature of acquired immunity and the secondary response or the anamnestic response. Okay. So that's why primary and secondary immune responses are carried out with B lymphocyte and T lymphocytes. Means if you are asked which cells are responsible to perform, so two very specific cells, B lymphocyte and T lymphocytes. So these are primary cells for acquired immunity. Okay. Now if you talk about B lymphocyte, they are responsible for producing antibodies. B cells are responsible for production of antibodies, and which are antibodies. Antibody means these are proteins to fight the pathogens, right? And T lymphocytes, they will be helping the B cells to produce the antibodies. So this is how your acquired immunity works for you, and the immunity you will be getting by B lymphocyte. The immunity you will be getting by B lymphocyte, that is called antibody mediated immunity. Antibody mediated immunity. And the immunity you will be getting from T lymphocyte, that is called cell mediated immunity. Why it's so? As because I said that when a pathogen will enter into your body, suppose this is the pathogen. So this pathogen will produce some toxin. This pathogen will produce some toxin, and these toxins are called as antigens. Okay. Now in response to this antigen, this signal will be given to your B cells. The signal will be given to your B cells or B lymphocytes. Now, who will be giving signal to the B cells? T cells are responsible for giving the signal to the B cells, and once B cells are notified that we got an attack, we are under attack, B cell will modify into another kind of cell, we call them plasma cell. These are activated B cells, we call them plasma cells. Right, now they are preparing themselves to produce army of protein, and this army of proteins will look like this Y-shaped proteins, and we call them as antibodies. Now these antibodies gonna come into your bloodstream. This antibody will come into your blood and lymph, means inside your body fluid, and these antibodies will block the attack of these toxins, and that is the reason the immunity provided by your B cell, that is called as antibody mediated immunity. Also you can call it humoral immunity. Humoral immunity. Why antibody mediated immunity is called as humoral immunity? Meaning of humor, you need to decode. Humor means your body fluid, and these antibodies are released where? Into your body fluid, means into your blood and into your lymph. So blood and lymph together can be called as humor of your body. So that's why the immunity is called as humoral immunity. And why the immunity given by T cells are called as cell mediated immunity? Because directly these T cells are involved into your immune response. That's why we call cell mediated immunity when you are getting help from any T cell. Okay. Now we'll talk about antibodies. So what can be the definition of antibodies? They are soluble proteins into our body fluids produced by B cells or plasma cells. Okay. So they are protein in nature. Antibodies are protein in nature. Now let's talk about their types. Remember, there are 5 types of major group of antibodies are there, and the commonly these antibodies are called as immunoglobulins. Immunoglobulin, or you can also call them as gamma globulin, a special kind of protein. So short form is Ig, immunoglobulin. So IgG, IgA, IgM, IgD and IgE. So these 5 kinds of antibodies are produced inside our bodies. Remember this chronology, GUM-D. So IgG, the most abundant antibody. IgG, they're present in like more concentration, and the least abundant antibodies are IgE. So this is the rank of antibodies based upon their abundancy, right? So IgG, the highest concentration of antibodies will be finding inside your body. They are IgG, then IgA, then IgM, then IgD, then IgE. Okay, and which antibodies are smallest and largest? IgG, these are the smallest antibodies, and IgM are the largest among antibodies. IgG are the smallest among antibodies, and IgM, they are the largest antibodies. Okay, what is the function of IgG? The normal antibodies always present into your bloodstream, that is anti-A or anti-B, which are responsible for blood grouping. They are IgG type, they are IgA type, specifically the Rh antigen, if you will be getting this Rh antibodies, Rh antibodies. When we were discussing about the disease called erythroblastosis fetalis, you know, there is a condition when mother is having a negative blood group and the baby is having a positive blood group. Then second baby onwards gonna get this disease called erythroblastosis fetalis, because during the time of delivery of first child, blood of the positive blood baby went inside the mother, and she is having negative blood group. So her body will be developing Rh antibodies, and these Rh antibodies are responsible for the disease called as erythroblastosis fetalis. So these are IgG kind of antibodies, and as because these are smallest, they can cross the placental barrier and harm the baby. IgA, these are the antibodies present into mother's milk or specifically colostrum, right? IgA also present into saliva, right? Okay. IgM, they are the largest among antibodies, and when in our body IgM is produced after a fresh infection. When there is a new infection inside your body, first your immune system, first your B cell will be producing IgM. Okay. IgD, remember, these are the antibodies which are present on the surface of B lymphocyte, and IgE, very important for us, these antibodies are produced during the time of allergic reaction. So allergic reactions, during the time of allergic reaction, actually it's your body which is producing IgE antibodies. And normal structure of antibodies are displayed like this: H2L2, means each antibody molecule is having two light chain and two heavy chain. So these two, white two are the light chain and yellow, these are the heavy chain. So we can write in this manner: this is L1, this is L2, this is H1, this is H2. So two heavy chain and two light chains are present into antibodies structure. So that's why they are written as H2L2. And obviously if you observe carefully, you are going to get variable part of heavy chains and light chain and constant part of heavy chain and light chain. Okay. Towards the upper end, you are going to get the variable part, and towards the lower end, you are going to get the constant part of the chains. Means the lower portion is similar for all the antibodies, but the upper end is dissimilar, it's different for different antibodies. So that's why upper part of these chains are called as variable zone, and the lower part of the chains, that is called as constant zone. Okay. And these variable zones are zones have one more importance: they have site for antigen binding. So they have site for antigen binding. So basically when a antigen will come to bind with the antibody, so they will be binding with this variable zone. So that's why these variable zone are also important, because they contain antigen binding site. So this is antigen specific antigen like lock and key, they will be binding with specific antibodies. Okay, now can you tell me which of the following antibody is present in colostrum? IgG, IgA, IgD and IgE. Can you write down in the answer in the comment section? Okay, so I can see the answer. Amrik Bashnoi has answered is IgA. Okay, so right answer. And Prabhakaran has answered as option 2. Okay, so both of them have given the right answer, IgA only. Okay. Now, if we draw the diagram of antibody in a large format, so the white two are the heavy chain and yellow two are the light chain, and these are the variable regions. Okay. Now we need to talk about few more information about the structure of antibodies. Okay. Now we know that this zone is variable zone, this zone is constant zone. Now if we are asked that if we talk about the linear chain of a protein is having an N terminal and a C terminal, and we know that each heavy chain and light chain, they are also chain of polypeptide, chain of amino acid, multiple amino acid. So where there is C terminal and where there is N terminal, so these ends are basically N terminal of protein chain, and these terminals are the C terminal of protein chain. So towards the variable region, we will be getting N terminal. At the end of constant region, we will be getting the C terminal of antibody molecule. Now the next question is asked that these antibody, these chains are interconnected with each other. How they are interconnected? They are interconnected with sulfur-sulfur bond, that is disulfide bond, right? How many disulfide bonds will be present inside this structure? So you can give the answer. There will be total 16 disulfide bonds. Okay. Now let's check those disulfide bonds. There will be 4 interchain disulfide bond. There will be 4 interchain disulfide bond. 1, 2, 3, 4. 4 interchain means disulfide bond which is connecting one chain with another chain. It is called interchain. And there will be, if you minus 4, there will be remaining 12. So there will be 12 intrachain disulfide bond present into this structure. So there will be 12 intrachain disulfide bond present into this structure. One, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 and 12th intra-chain disulfide bond. So you need to remember this very important information about the structure, basic structure of antibody. So total there are 16 disulfide bonds you are gonna get inside this structure, and out of these 16, 4 are interchain disulfide bond and 12 are intra-chain disulfide bond. Okay. Now, let's move forward to the next topic, that is active immunity and passive immunity. Okay. So now if we check the under section, or if we check the subgroups of acquired immunity, we can divide into two groups, active and passive. So active immunity means it is the immunity in which antibodies are produced in a host body when the host is exposed to antigens, living or dead microbes or other proteins. It's a slow process, so it is produced by two ways: natural active immunity and artificial active immunity. So it's very basic. What is the difference between active immunity and passive immunity? If you are the host and you inside your body only you can produce the antibodies against the pathogen, so that will be called as active immunity. And if you are sick, you are diseased, you are infected, and you are taking antibodies from any other human being or from any other animal already produced. If you are taking ready-made, if you are taking ready-made antibodies from other human or from other animal, then that mode of immunity is called as passive immunity. Okay. So we were talking about active immunity. Let's explore what is natural active immunity. So it is developed during the time of natural infection by microbes. So when you are infected by any of the pathogens, not normally or naturally your body will be producing antibodies against that. So that is example of natural active immunity. What is artificial active immunity? It is developed by injecting the microbes deliberately during immunization. Simply, if we talk about vaccination. So what is OPV, oral polio vaccine? You are taking one drop of oral polio vaccine and that drop is containing some inactivated or dead polio virus. When you are taking the dead or alive viruses into your body, so your immune system gonna create a response and produce antibody. So vaccination is example of artificial active immunity. Okay, now let's talk about passive immunity. Already I said that here ready-made antibodies are directly given to the body. It is also two types: natural passive immunity. Now what is natural passive immunity? When antibodies from mother through placenta enters into the fetus, or when through during the time of breastfeeding IgA antibodies are entering into the body of the child. So that is example of natural passive immunity. Okay. Now what is artificial passive immunity? It's anti-tetanus serum or anti-venin injection, anti-venin serum which is given against snake bite. Okay. So if you are a doctor and suddenly saw a patient came to you which is bitten by a snake, then what you will be doing? You will be injecting anti-venin serum. Now what do you think that anti-venin serum is containing? The anti-venin serum is containing antibodies to neutralize the toxin of that particular snake venom. And where this anti-venin serum is produced, they are produced inside, those antibodies are produced inside the body of rabbits or horse by injecting low dose of snake venom inside their blood circulatory system. So when a doctor is administering anti-tetanus serum or anti-venin injection, tetanus serum, anti-tetanus is given against tetanus, anti-venin injection is given against snake bite. So both of them are containing prepared antibodies, right? So that is why we are telling the mechanism of artificial passive immunity. Now let's talk about vaccination and immunization. So remember, vaccination and immunization, they are not passive immunity, they are artificial active immunity. Okay. So these are based on memory of the immune system. Firstly, we will be talking about vaccination. So vaccinations are always active immunization. In this preparation of vaccine or antigenic proteins of pathogen or inactivated pathogen is introduced into the body. So it will be resulting in the development of antibodies. Now, prior to any infection, before any infection, as a precaution, we are injecting the real pathogen. Obviously, the pathogen is not much virulent, it's attenuated or it's heat killed, and then we'll be inserting that inside the body of a healthy human being, and his or her immune system gonna make antibodies, right? And we know that the memory capability or the anamnestic response of acquired immunity. So when in real world a real pathogen will attack that particular person, the immune system inside the body is already prepared with the antibodies. So that is the importance of vaccination. Okay. So during actual infection, the antibodies neutralize the antigens. So the toxins produced by the pathogens, we call them antigens. Okay. These vaccines are generate memory B and T cells, and they recognize the pathogen quickly. So that's why polio vaccine, hepatitis B vaccine, DPT, diphtheria, pertussis, tetanus vaccine are some example. So when somebody is administered with polio vaccine, specifically OPV, oral polio vaccine, the 1 or 2 drops, they are basically containing heat killed or dead polio virus. If you are injected DPT vaccine, diphtheria, pertussis, tetanus vaccine, then that injection is containing the these 3 pathogens: diphtheria, bacteria for diphtheria, cornybacterium diphtheria, bacteria for pertussis, and bacteria for tetanus. Okay, Clostridium tetani. If you are getting vaccine of hepatitis B, then actually you are getting the injection that is containing hepatitis B virus in attenuated format, right? Okay. Now, vaccines are produced using DNA recombinant technology, that is hepatitis B vaccine produced from yeast, that is a second generation vaccine. So what we are doing, by we are making a genetically modified yeast. We are making a genetically modified yeast by inserting the gene setup from this hepatitis B virus, and inside yeast that antigen is produced. Now we will be purifying the antigens from the body of yeast and will be injecting into the body of human being, and that's how we are immunized. So you need to remember, hepatitis B vaccine, which is a second generation vaccine that is produced by taking help of biotechnology, that is with the help of recombinant DNA technology or DNA recombinant vaccine. Now, passive immunization. Now, if you talk about passive immunization, they are simply the anti-venin serum or anti-tetanus serum. So, it is direct injection of preformed antibodies or antitoxin. It is required for quick immune response. See, for active immunization or vaccination, we have a lot of time that our body will be producing antibodies. But if there is a snake bitten patient, you don't have time to produce antibodies inside the body. So that's why for quick action we have to inject prepared or ready-made antibodies or antitoxin inside the patient's body. So that's why immunization against the tetanus or snake venom is example of passive immunization. Okay. Now the next topic is allergies. So it is the exaggerated response of our immune system to certain antigens present into the environment. Okay, and those certain uh, some antigens which are generally will not be harming normal person, but will be posing a threat to some particular allergic individuals, and we will be calling those antigens as allergens. like pollen grain. Generally, if I smell a flower, I won't get allergy. But if my sister or my brother, when he is smelling a flower, the next hour they are getting allergic reaction inside their body. So the pollen grain, the similar pollen grain is not posing any threat to my immune system, but activating their immune response. So that's why pollen grain is acting as allergens for them. So substances causing allergy, that is mites present in dust, pollens, animals, dander or fur of animals. So what happened? Antibodies produced against the allergens are already we have discussed, that is we are knowing there is IgE type. IgE, their antibodies responsible for allergic reaction. Now this Ig will bind on mast cell to release chemicals like histamine and serotonin, and then what will happen? It will result allergic reaction. So basically how it works inside your body. So suppose this is pollen grain of a flower. This is pollen grains of a flower. For an allergic person, the pollen grain will enter into the body, and as a response, your body will start producing IgE antibodies. Now this IgE will go and bind with the surface, of mast cell, right? Now this mast cell will get activated and will start releasing histamine. Okay. Now this histamine is a potent vasodilator. What do you mean by vasodilator? If diameter of your blood vessel, previously it was like this is the diameter of your blood vessel. Upon the action of histamine on it, the diameter will be increased. So that is called vasodilation. And when blood vessel is dilated, then phagocytic cells or immune cells will come out from your bloodstream, and they will try to invade into your tissue, causing inflammation and allergic reaction. Now symptoms will be like this: sneezing, watery eyes, runny nose, difficulty in breathing. Now why there will be difficulty in breathing? As I said, this histamine secretion will cause vasodilation but bronchoconstriction. It will choke your trachea and bronchi, this histamine. And when this trachea and bronchi are getting choked, then what will happen? You will face difficulties in breathing, right? Wheezing sound will be produced. You know about asthma or allergic asthma, right? Or sometime there will be formation of itching skin or skin rashes. Now how it is treated? Drugs like antihistamine or adrenaline and some steroids are there to quickly reduce the symptoms of allergy. So generally antihistamine like histaminidase enzymes are given so that the produced histamine is neutralized. Okay. Asthma is a respiratory disease due to allergy. Okay. Now another immunity related disease called as autoimmunity. What is autoimmunity? In higher vertebrates, the memory based acquired immunity evolved based on the ability to differentiate in between foreign organism from self cells. Specifically, these are the function of your T lymphocyte, right? They are important elements of acquired immunity, and we know that the cells of acquired immunity, they are very much specific, like in term of choosing their antigens and the pathogens. So they can differentiate between your own cell and the pathogenic cell. So this is the normal capability of our immune system, and that is produced inside our body through a process called as clonal selection. Like if you are preparing for NEET, then you are not a doctor. You have to crack the NEET exam, then only you will get admission into medical college, and after completion of your fifth year, then only you will become a doctor. Similarly, when your body is producing T cells, right, T cells are produced by your bone marrow, and T cells are matured into your thymus gland. So inside your thymus, what happens? Your body uh, is taking examination of those immature T cells, and if T cells are able to pass the examination, your body is having its own way, right? Then only those T cells are recruited as our acquired immunity cells. And if they fail to do so, then it's the duty of your body to kill them or to deactivate them. But sometime there will be some mismatch during the time of this clonal selection of your healthy T cells. So what happens, sometime due to some genetic and other unknown reasons, maybe some certain some sudden mutations are happening into your body, the body will be attacking self cells, resulting damage of your own tissue. So it is called as autoimmune disease, and one of the example is given as a rheumatoid arthritis. So what happens in case of rheumatoid arthritis? The rheumatoid factors, your own modified antibodies, these are kind of IgM. These rheumatoid antibodies will be attacking your joint, the joint between two limbs. This particular joint is called as a synovial joint, and this synovial joint is having synovial fluid and synovial membrane, so that while there will be movement, the shock will be absorbed by that synovial fluid. So what will happen here in case of rheumatoid arthritis? This modified antibodies will destroy the synovial membrane and synovial fluid, and hereby your body will face in case of movement, and there will be pain, immense pain in your joints, and that condition is called as rheumatoid arthritis. Okay, few children may face hay fever. That's also sorry, that's an allergic reaction. Autoimmunity, one another disease called as Hashimoto's disease, right? This is one of the autoimmunity or disease found into our body. Then lupus erythematosus, another autoimmunity disease found into in our body for some person. Okay. So the human immune system consists of lymphoid organs, tissues, cells, and soluble molecules like antibodies. So remember this sentence: antibodies are soluble into your plasma. They are soluble proteins. Okay. Now we'll be focusing on lymphoid organs. Now what do you mean by lymphoid organs? The special organs of our body where maturation and proliferation of lymphocytes occur. Who are lymphocytes? B cells and T cells. B cells and T cells are called as lymphocyte. The site of production and site of maturation is called as lymphoid organs. So there are two kinds of, yeah, after this topic I will be giving 5 minutes of break. After this topic, this lymphoid organs, yeah. So there are two kinds of lymphoid organs are there, primary lymphoid organ and secondary lymphoid organ. Now what do you mean by primary lymphoid organ? The organs where lymphocytes are matured and differentiated to antigen sensitive lymphocytes, where B cells and T cells are produced. They are called as primary lymphoid organs. And who are they? For us, primary lymphoid organ means our bone marrow. Inside our bone, we have bone marrow and our thymus gland, a gland near to our heart, right? So these two glands, these two organs are called as primary lymphoid organ. These are responsible for formation and maturation of B lymphocyte and T lymphocyte. Okay. Now, what is secondary lymphoid organs? Now, secondary lymphocyte organ, the organs to which matured lymphocyte migrates from the primary lymphoid organs and interact with antigens and then proliferate to become the effector cell. So again I am explaining it. So if you want to become a soldier, then what you have to do? You have to prepare for the exam, right? Then after qualifying the exam, you have to go through a training schedule, very hard training seasons will be there, training schedule will be there, and if you qualify those trainings, then only you will be getting a gun, and during the time of war you have to use those guns, right? Similarly, for our immune system, the soldiers, we need to produce immune soldiers, and they are T cells and B cells. Where they are born inside bone marrow and thymus, right? So the house where they are born, that house is called as primary lymphoid organs, right? Okay. Now after that, what will happen? Your parent will be sending you to school, will be admitting you to school, and inside your school you will be interacting with your friends, you will get to know about the real life, what's happening in the world. Then you will be preparing for the exam. Some of you will become doctors, some will become engineers, you will become teachers, some of will become IAS, some of will become businessmen, right? So similarly, after a specific stage of development, these T cells and B cells will be sent inside the secondary organs so that they can interact with the antigens, which are antigens, the toxins produced by the bacteria virus, and they can choose their journey, they can choose their pathway, that what they want to become, right? So that's why it's written, the organ which mature lymphocytes migrates from primary lymphoid organs and interact with antigen and then proliferate means they are dividing, cell division is happening to become effector cells, right? So effector cells means now these B cells and T cells are ready to fight with the infection. Okay, and which organs are considered as secondary lymphoid organs? They are spleen, the graveyard of RBC. Spleen is also called as the blood bank of your body. Why spleen is called as graveyard of RBC? Because when RBCs are expired after 120 days, right? The lifespan of one RBC is for 120 days. Then they will become bigger in size. Spleen has some macrophage. So they will be trapping those enlarged RBC and will be killing them. So that's why we call spleens as graveyard of RBC. Lymph nodes of our body, tonsils, Peyer's patches and mucosa associated lymphoid tissue, malt and appendix. So these are part of your secondary lymphoid organs or secondary lymphoid tissue. Now remember the MALT, mucosa associated lymphoid tissue. Now what do you mean by mucosa? These are the wall. Mucosa is a special tissue layer present into the wall of your alimentary canal, into your respiratory passage, or into your gastrointestinal as well as reproductive passage. So located within the lining of respiratory, digestive and urinogenital tract, and it constitutes 50% of all lymphoid tissues. 50% cells of your, if your overall, if you have 100% lymphoid tissue into your body, the 50% of the cells will be belonging from the group called MALT, mucosa associated lymphoid tissue. Okay. So now I'm giving 5 minutes of break, and after that I'll be coming back. Okay, so now we'll be starting the next topic, that is HIV AIDS. So till now, whatever the topics taught for today, if you have any question or if you want to ask, you can write down your question or query into the comment section. After that, we'll be moving forward to the next topic from this chapter, that is HIV AIDS. Okay, so when you talk about this particular disorder or syndrome, the full form is acquired immunodeficiency syndrome. Okay, now what is syndrome? Why it is not called a disease? Because there will be a group of symptoms of multiple diseases will appear if somebody is suffering from this particular condition. So that's why rather calling it a disease, we call it a syndrome. Symptoms of multiple diseases. And why it's so? Basically, the virus called HIV virus, human immunodeficiency virus, that will be attacking the backbone of your immune system. They will be destroying the T helper cell, the most crucial cell to maintain your acquired immunity. T helper cell, a kind of T cell, right? So when T helper cells are getting reduced, even simple or even normal pathogens will also attack you, and as because you don't have any sort of immunity or immune cells inside your body cannot produce antigens against that. You will be, your body system will be submitted and you will die gradually. Okay, so the disease was first reported in the year of 1981, and you know that disease was contaminated or from into human being from monkey. Yes, and this disease killed more than 25 million people over last 25 years. Yes, and its characteristics by decreased number of T cells, particularly decreased number of T helper cells. A cell which is required for activating the B cells so that B cells can produce antibodies for us. Okay. Now the name of pathogen is HIV and full form is human immunodeficiency virus, because after the attack you are gonna be deficient in terms of your immune cells or acquired immunity. What kind of virus is this? This is a kind of retrovirus. Now what do you mean by retrovirus? See, the genetic material of this virus is double stranded, a single stranded RNA. The genetic material of this virus will be single stranded RNA. Now what happen inside your body? Generally your normal genetic material is double stranded DNA. By the process of transcription, DNA will be by the process of transcription, mRNA is produced from DNA, and from mRNA with the help of ribosomes your body will be producing proteins. See, so this is called as central dogma, right? So the process of formation of DNA to mRNA, this is the modern process. But if you are forming mRNA to RNA to DNA, the process will be called as reverse transcription. Reverse transcription. And this virus, HIV virus, can perform reverse transcription into the body of host which is attacked by HIV virus. So that's why we call them as retrovirus. They are not forming DNA to RNA. They have the capability to produce RNA to DNA, right? So that's why the retro word is used. A primitive configuration. So it's a kind of enveloped enclosing RNA genome is there. Enveloped means outer protein covering will be there. That's why we are naming these as enveloped virus. And what are the major components you are gonna get inside this enveloped virus? There will be two identical single stranded RNA. Single stranded RNA will be there. How many strand of two single strand will be there? Two single strand will be there. Okay. Two reverse transcriptase enzyme will be there. This reverse transcriptase enzyme is necessary to perform reverse transcription, right? For this method only, this virus, they got the name retrovirus. There will be two integrase protein inside and there will be two protease enzyme. Now let's check the structure of HIV virus. So this is the core of virus, and in the core you can observe these two are the identical single stranded RNA. So the core of the virus is made up of two single stranded RNA, and in the core region you are getting this two reverse transcriptase enzyme as well. Reverse transcriptase enzyme which is necessary to form RNA to DNA. The process of reverse transcription. Okay, these two are protease enzymes and these two are integrase enzyme. Now what is the importance of integrase here? The virus will just inject the RNA strands into our cell. Now after that what will happen? The viral enzyme will convert RNA into DNA. Now this viral DNA will be incorporated or merged with our DNA or our genome, and for that merging process they need this integrase enzyme for integration of viral DNA with our original genomic DNA, right? Okay, and that core is surrounded by one covering, protein covering called as inner capsid. Capsid, the name of that protein covering, right? And over this protein capsid or the inner capsid, it has two phospholipid bilayers. It has two phospholipid layers or phospholipid bilayers, and some spikes are projected within this phospholipid bilayer. And what are the importance of these spikes? These spikes are basically the sensory area of this particular virus through which it will be recognizing the host cell surface receptors and seeking permission to inject the genetic material inside the host cell. So these are the functions of this spike. Okay, and you can see here these are the different kind of spike protein you will be finding. This part is made up of glycoprotein type 120. This part is made up of GP41, that means glycoprotein kind of 41 kind of protein. Okay, GP41 and GP120. They are glycoprotein molecule, and we call them as spike. What is the importance of these spikes? If this is the host cell, the host cell membrane will be displaying some surface receptor molecule. Now, if you want to enter into the cell, if you want entry inside the cytoplasm, then you have to satisfy this surface receptor present on the host cell. Then only if there is a match in between the receptor and the molecule, then only the host will create a passage and will give you a yes signal that you can enter. So, what happens here? This virus will come closer to the cell, and with the help of this spike protein, they will seek the entry, and as because this viral spike protein, they satisfy the pattern of the host cell surface receptor, so easily the host will be allowing or giving permission to the virus to inject the genetic material, that is a single stranded RNA inside the cytoplasm. Okay. Now, the later on the attack to the host cell will be done by this product of this single stranded RNA, which was injected by this particular virus. So, that is the importance of the spike proteins. Okay. So, this HIV attacks CD4 cells. So, it is very much specific. It can only satisfy some specific cells with specific receptor and CD4 kind of receptor. If the cell is having CD4 kind of receptor, then only we will be calling those cells as CD4 plus, a special kind of lock. Now, if you have the key to open the lock, then only you can enter into the room. It's something like that. So, which all cells are having this CD4 receptor on the surface? T helper cell, which is the primary target for HIV, right? And also macrophages. Few of the macrophages also will be exhibiting CD4 receptor in their cell surface. Okay. Now, these macrophages are called as HIV factory. Now, simultaneously, when this virus will be entering into your bloodstream, they will attack T helper cells, they will enter into the macrophages, but they will kill T helper cell, they will not be killing macrophage. It's a kind of friendship they are making with macrophage, as because they will be hijacking the central dogma of macrophage. They will be hijacking the genome of the macrophage and utilizing it to produce more and more HIV virus. Okay. So, that is why we call macrophages as HIV factory, and that is why HIV virus will not give a threat to macrophages, but will kill T helper cells. Okay. So, HIV kills T cells but do not kill macrophages. Progressive decrease in T lymphocyte causes, now gradually if T cell count is going down, then that will cause bouts of fever, mean multiple times episodes of fever will be there, diarrhea will be there, and gradual weight loss will be there. That is why another name of AIDS is slim disease. Okay. Gradually you will be becoming thinner. Okay. Now, severe causes. Severe cause means full blown, when there is very less count of T cell into your body, and the CD4 cell means T helper cell count, that is less than 200 into 10 to the power 6 per liter volume of your blood. So, what will happen? Multiple opportunistic pathogen will attack you. when CD4 cell means T helper cell counts are too much low due to multiple attack of HIV virus, then what will happen? Opportunistic pathogen will take the advantage, as because you don't have your immune system is not working properly. So in that case, you will be affected by tuberculosis, which is caused by mycobacterium. You will be affected by toxoplasmosis, which is caused by toxoplasma. You will get multiple kind of fungal infection, and possibilities are cancer of skin and lymph nodes will be taking place. Okay. Now, incubation period for HIV virus. Now, what do you mean by incubation period? The day the virus has entered into your body, that is called as day of infection, and for few days or few months or few years, it will be increasing the population inside your body, but it will not pose any threat, it will not cause any disturbance for your normal biological function. It will remain silent, and one day it will become virulent, it will become active, and it will start destruction inside. So that we will be calling that symptoms are appeared. So from entry to appearance of symptom, that particular time span is called as incubation period. So infection to appearance of symptoms, that time span is called incubation period, and for HIV virus from few months to many years, like 5 to 10 years, as long as 10 years can be the incubation period. The patient may remain asymptomatic for 10 years. Okay. Now this is spread through, how it's spread through body fluids or mucosa to mucosa connection. If there is transfer of blood or if there is connection of mucosa of two human being. Now how it's gonna take place? Okay. It gonna transmit during the time of sexual intercourse. That's why HIV AIDS is considered is one of the most dangerous and non-curable STI, sexual tract infection, or sexually transmitted disease, or venereal disease, because during the time of sexual intercourse, when two partners are doing coitus, then there will be mucosa to mucosa connection, which is present into the vagina and on the upper surface of the penis, right? On the inner surface of penis. So that mucosa to mucosa connection will perform fluid exchange, and that fluid exchange will transmit that virus from one partner to another partner. There can be transmission of this virus through blood. Transmission can occur from sharing infected needles, or from infected mother to fetus or child. How the disease will be transmitted from mother to fetus? Through placenta. This virus can cause a cross placental barriers. How this disease will be transmitted from mother to child? During the time of breastfeeding. Okay, but here we will be talking about one misconception, that this disease do not spread through cough, touch or physical contact or mosquito bite. Okay, so suppose a person is suffering from HIV AIDS and a mosquito is biting. So you will be thinking like malaria, like dengue, the mosquito can transmit the disease, but that is not possible, because when a mosquito is biting or sucking the blood of HIV patient, the HIV virus won't gonna survive inside the mosquito's gut, because mosquito's gut is too much alkaline, and that much alkaline is not good for viability of HIV viruses. That's why mosquito bite cannot transfer this disease. Okay, or simply if one man is touching another, or one partner is kissing another partner, so in the time of kiss, in the time of normal touch, or in the time of mosquito bite, there is no chance that disease can be spread through. Okay. Now, people with high risk of getting this disease, remember, if a person is having multiple sexual partners, okay, intravenous drug abusers, a group of friends, suppose one got this HIV AIDS and without sterilizing with the same syringe, they are injecting drugs into their body. Now, from one patient, with the surface of the infected needle or injection during the time of intravenous drugging process, this can be transmitted. Okay. Repeated blood transfusion, like a person, if the person is having thalassemia, SCA, sickle cell anemia or bone marrow diseases, they need to take blood frequently. Okay, so if it's not cared properly during the time of blood transfusion due to the infected needle or due to like after upon taking the infected blood, the disease may get transmitted. Okay. So, it's widely used. I will be talking about diagnosis, how HIV AIDS can be diagnosed. So, widely used method of diagnostic test, that is also called as screening, is done by the process of ELISA, enzyme linked immune sorbent assay. So, it's a preliminary detection, but this is not the confirmatory test. So, what is the confirmatory test of HIV AIDS? That is Western blotting technique or HIV RNA PCR. Okay. Now, what is the difference between confirmatory test and screening test? Okay. Now, confirmatory test means there is no chance the report you may get, that is false positive or false negative. Sometime ELISA will give you false positive or false negative result. Means ELISA test result is not 100% reliable or 100% accurate, but it's quite cheap and easier. So, that's why a preliminary ELISA test can be done. But if you want to confirm about this disease, then you must have to check for Western blotting technique and HIV RNA PCR. Okay. And what about the treatment? Though this is non-curable disease, but if you take some antiviral drugs, okay, for some time the cues or the symptoms can be suppressed or reduced by taking antiretroviral drugs like zidovudine, stavudine. There are very few antiretroviral drugs are there. Okay. Now, these drugs only can prolong the life of patient, but death is inevitable because this is non curable at all. So, by taking proper precaution and drugs and treatment, your lifespan can be enlarged for few months or few years, but death will come for sure. Okay. That's why it said that AIDS has no cure, only prevention. So, that's why don't die of ignorance. That means before marriage, do the blood test, do the confirmatory test, either your partner is having HIV AIDS or not, while you are taking injection or while you are doing transfusion of blood. Okay. Do a proper cross check, either the blood or the injection needle is free from contamination or not. Okay. Now, what is NACO, National AIDS Control Organization? It's an organization which is responsible for tracking the ongoing condition of HIV AIDS infection in our country. Now we are done with HIV/AIDS. If you have any question, if you want to ask any question from the previous topic, that is HIV/AIDS, you are free to ask. You can write your question into your comment section, and the next topic we are going to start, that is cancer. So before starting the topic, if you have any question from the previous topic, that is HIV/AIDS, or from immunity, you can write your question into the comment section, and happily I will be giving you an answer. So no question from anyone. So I am moving forward to this topic now, cancer. Now before moving, let me once summarize the important infos about HIV/AIDS. So again, I am repeating once. HIV is caused by retrovirus. What is retrovirus? It's a kind of a single-stranded RNA virus, right? Why HIV virus is called as retrovirus? Because while it is affecting its host, like our cells are host cell for HIV virus, and when it's injecting the genetic material as single stranded RNA, like after injecting, it has the capability to change the RNA into DNA format. So it can change, it can change the RNA to DNA, that is called as reverse transcription. It can perform reverse transcription. That's why HIV virus is called as retrovirus, right? If an organism is having the capability to perform reverse transcription, then it's kind of retro, because generally in what is opposite to retro? Retro means primitive, and opposite is modern. So all the modern organisms, all the eukaryotic cells and prokaryotic cells, they can only change DNA into RNA, right, by the process of transcription. That's why bacteria, fungi, animals, plants, protozoa, they are not retro, but few viruses are retro in nature. Okay. So replication of retrovirus. Yes, replication is of retrovirus will be like this. If this is the cell, if this is the cell, and this is the nucleus. So suppose this is the host cell. So the virus will enter. This is the HIV virus. Sorry, the virus will not enter, but it will inject the single stranded RNA along with the reverse transcriptase enzyme, integrase enzyme, and protease enzyme, right? So what will happen next? This reverse transcriptase enzyme will convert this RNA molecule into DNA. It convert this RNA molecule into DNA, right? Now this is which kind of DNA? This is viral DNA. This is DNA of virus. Now the integrase, now this is the duty of integrase enzyme to integrate the viral DNA, to take the viral DNA with the cellular DNA. It will merge the viral DNA with the cellular DNA. So now this viral DNA is the part of cellular DNA or the part of genome. So what it will do? It will influence the central dogma of host cell. It will hijack the central dogma. Now the host cell will not produce protein which are required for host cell, but host cell will be producing proteins which are good for which are required for virus production. So the host cell will produce viral mRNA again, and this viral mRNA with the help of ribosomes will be producing viral proteins. And if viral proteins are produced, so inside the cell this new miniature viruses will be replicated. That means this is how, by utilizing the host cell as a machine, the virus will produce its progeny, right? This is called virus multiplication or viral replication. So when there will be formation of hundreds or thousands of virus inside the host cell, all these virus want to come out. So what they will do, they will rupture the host cell membrane or host cell, they will blast the host cell, and gradually they will be coming out and they will be attacking a new cell. So this is how the HIV virus will be attacking a particular host cell and forming multiple copies of it. Then after destroying the host cell, it will attack some new T cell. So this is how it continues its journey. Okay. So still, if you have any uh, question from HIV/AIDS, you can ask. You can write down in the comment section. Then we will be starting the next topic, that is cancer. Okay. Then let's start. So the major, we know that cancer is non-infectious disease and that is major cause of death. Okay. So if you are asked, tell me about a disease which is non-infectious in nature and causing a lot of death, then answer will be cancer. Now if you are asked, tell me name of an infectious disease which is very much fatal in nature, then you have to answer, then that is HIV/AIDS. Okay. Now for cancer, more than 1 million Indians suffer from cancer and a large number die from it annually. Okay. In 1 year, near about 8 lakh people, 8 lakh people will be dying due to cancer, and we need to study oncogenic transformation of cells. Now what do you mean by oncogenic transformation? See, oncogene means cancer causing gene. Oncogene means cancer causing gene. Generally, these genes are present into silent mode, into switched off format, into normal cell. But when these oncogenes are getting switched on, getting activated, then the cells will be called as oncogenic cell, and that can cause malignant tumor, right? And whenever there is formation of malignant tumor into our body, the condition will be called as cancer, means uncontrolled growth of cell of our body. It can be bone cancer, it can be bone marrow cancer, it can be blood cancer, it can be breast cancer, it can be cervical cancer, it can be any glandular cancer. There are different kinds of cancer. It can be prostate cancer. So, a particular tissue cells are showing unlimited growth or unlimited proliferation. So, that is the meaning of oncogenic transformation. Is not maintaining any limit. Naturally, what happens that in your body, normally a single cell, a cell will be dividing for 50 to 60 times. Then the whole set of cells will be will die, and a new set of cell we call stem cell will be replacing the old cell. So, that is how total cell number is maintained into our body, right? But if there is any variation, if there is excessive growth of a tissue, that condition is called as tumor. Now, you may get two types of tumor. One is benign tumor, another is malignant tumor. Now, benign tumor, excessive growth of cell is observed for a particular tissue, but within few days the body will reduce the growth rate and there is no more enlargement in that tumor. Now, that tumor has become quiet. So, we will be calling that as a good tumor. Benign means somehow good. Now, if there is formation of a tumor or excessive growth of a cell mass and somehow it is not possible for your body design to check the growth. So, there is unlimited, the cells are growing like anything, that is out of control. So, that dangerous, that bad tumor is called as malignant tumor and the condition is called as cancer. Okay. So, how these oncogenes are activated? Just now I was telling about oncogenic transformation of normal cell. Okay, these normal cells are activated by the influence of some agents and we call them as carcinogens. What do you mean by carcinogens? Cancer causing agent. Cancer causing agent. Okay, those who can influence your cell to become this oncogenic, those who are responsible for those oncogenes to be activated. Now, these carcinogens can be physical carcinogen. Now, what do you mean by physical carcinogen? They are the radiation. They are different types of radiations. We will be considering them as physical carcinogens. Now, radiation can be of two types. Radiations can be of two types: ionizing radiation and non-ionizing radiation. Now, what do you mean by ionizing radiation? X-ray and gamma ray. X-ray and gamma ray, these radiations are called ionizing. Why these radiations are called ionizing? Because when you are bombarding the X-rays or gamma rays on the surface of one atom or molecule, it has ability to displace the electrons. So, when electrons are displaced from the outer shell, outer orbitals, then what will happen? That atom will get converted into an ion. So, that is why these radiations are called as ionizing radiation. And UV ray, ultraviolet rays, these are considered as non-ionizing radiation. They do not have that much energy to displace electron from the surface of an atom. That is why UV rays are considered as non-ionizing. But if you are exposed to UV ray, X-ray or gamma ray, you may get cancer, right? Okay. Generally, when you watch movies, We have seen exposure of these gamma rays have produced superhumans like Hulk. But in real life, what will happen if you are exposed to gamma rays? You will die due to cancer. Now, let's talk about chemical carcinogens. Now, chemical carcinogens are present inside tobacco smoke, right? Now, generally, if you are asked that this smoke produced while you are smoking bidis or cigarette, and that tobacco is containing nicotine. Nicotine is not responsible for cancer. Rather, this tobacco smoke contains some polycyclic aromatic hydrocarbons, in short form we call it PAH, some aromatic amines, some nitrosamines, then some benzene, and these aromatic components, or these aromatic hydrocarbons are responsible for activating the oncogenes of your body. It's not nicotine, remember. Okay. Then some biological carcinogens. Biological carcinogens are oncogenic viruses. Now, how they will be affecting you? Few virus like papilloma virus, few species of papilloma virus, right? They can switch on when they are attacking you. They can incorporate oncogene, activate your oncogene, and then you may get cancer. Okay, so those oncogenes are called viral oncogenes. When your genome is getting oncogene from a viral infection, right? So those oncogenic viruses acting as they're acting as oncogenic virus or carcinogenic biological agents. Okay, you have some cellular oncogenes already present into your genome, right? No, but normally they are present as switch off format. They are inactivated, but once they are activated spontaneously or by any influence of some chemical, some physical physical radiation or some chemical agent, right, or spontaneously if they get activated, then they will cause cancer into your body. So, when you are talking about biological carcinogens, you need to talk about two things: oncogenic viruses. When these viruses will be infecting you, your genome will be getting some cancer causing genes. Yes, Nagaraj, what you want to say? Welcome to the class. Do you want to say anything, Nagaraj? So, I was talking about oncogenic virus and cellular oncogenes. Now, what is cellular oncogene? Again, I am repeating. Cellular oncogenes, these are already present inside our genome, but they are not activated. They are switched off. But due to some infection or due to some influence of some physical radiation or some chemical agent, if they get activated, they will cause cancer or unlimited proliferation of our cells. Okay. So, now we will try to differentiate between normal cell and oncogenic or cancerous cell. So, for normal cell, see, these are the criteria can be observed. Cellular growth is highly controlled and regulated. As I said, that for a normal cell will exhibit cell division maximum 50 to 60 times. Then they will die and a new set of cell will replace them. So, they have proto-oncogene, and these proto-oncogenes are those switched off format, right? These are good actually. This proto-oncogene, they will be controlling growth and proliferation. They are responsible for controlling normal growth and normal proliferation. Now, normal cells have tumor suppressor gene. This tumor suppressor genes are responsible for uh, inhibition of proliferation. Okay, whenever a cell will try to divide in unnatural way or aberrative way, your tumor suppressor gene will inhibit it or it prohibit it, right? Normal cells have suicide genes. Now these are very much important. They are responsible for apoptosis or PCD, programmed celled death. PCD means programmed celled death. As I said that your cells are already by default programmed that after 50 times of division automatically they will die. That is influenced by suicide genes. Actually they are good for you. Okay. Now they will show contact inhibition. What do you mean by contact inhibition? If there is a certain given area, we have a limited area into our body, and suppose these cells, one cell started cell division. Now while number of cells are increasing, they will touch each other. Now they have sensitivity. If my zone or my surface is touching you, I won't be progressing into your zone. I will be limited to my area only. So that is called contact inhibition, right? That is exhibited by normal cells. Now normal cells will show telomerase activity, another very much important criteria. What do you mean by telomerase activity? Yes, uh, telomerase enzyme is crucial for maintaining controlled cellular growth. During every cell division, your telomerase enzyme is splicing or cutting a short portion of your DNA, and that's causing aging. That's why with time we are getting older. Our body texture is modifying time to time and causing aging due to the activity of telomerase activity. But for cancerous cell, what will happen? They will lose the telomerase activity, they will become immortal cells. Yes. Next, cell addition. That's what the another mode of contact inhibition. When a cell will when a group of cell will be formed, they are responsible to make a tissue, and the cells are attached by intercellular matrix, that is called cellular addition. They will be tightly packed together if we are talking about cells of normal tissue or normal cells or non-cancerous cell. Yes, obviously this is a very important chapter for biology, because if people after 1 or 2 years gonna become a successful gonna enter into a medical college, and after few years will become a successful doctor. So obviously you have to talk about diseases and all. That's why. Now let's check the properties exhibited by cancerous cell. So cancerous cell will exhibit uncontrolled growth. They lost the contact inhibition. Like if there is a confined region is there into your body, now if cells are modified into cancerous cells, they will try to overlap each other, as because they are dividing like anything. Technically they are not dying after 50, 60 or 100 divisions. So there will be overpopulation, and that overpopulated or excessive growth of cell is called as cancer or a tumor, right? So these conditions are called as neoplasia. Neoplasia. What is neoplasia? Uncontrolled growth of cells, and cells are losing contact inhibition, and they are ignoring apoptosis. They are not dying. That's why every single cell, cancerous cells, are immortal in nature. Though you are not immortal, you will die due to cancer, but if I take out, if you are suffering from cancer and if I take out your cancerous cell and culture in laboratory, they will not die under specific condition. But if I take your normal cell and if I culture into laboratory, they will die after certain time. Okay. There will be loss of cell addition. There will be tendency that cells will not follow the intracellular addition principle. They will start migration from one place to another place. There will be angiogenesis, means those cells, those group of proliferative cells will be calling blood vessels to give them nutrition. There will be metastasis. This is the most dangerous property of cancerous cell, as because they have lost the cellular addition, they are not attached with each other anymore inside an intact tissue. So the migratory cells will be moving with your blood circulatory system and will try to establish another tumor in some other place. Okay, there will be genomic instability will be there, and those cells gonna suppress your immunity. Even your immune cells are not able to kill them, and generally we can observe enlarged nucleus, right, due to their DNA replication is malfunctioning, or DNA replication has become erotic in nature. Now this is the difference between benign tumor and malignant tumor, and as I was saying, benign means somehow it's good, it will be there will be proliferation of cells first, but after some time they are gonna confined into a particular way, right. So that's why these harmless tumors are called as benign tumors. But if you talk about malignant tumor, they crossed the limitation. They have started migration from one place to another place. So they are dangerous, they are bad. That's why they are called as malignant. Okay. So the same thing is written here. Tumors are of two type. Benign tumor, benign means kind or good. Okay, they are confined to their original location. They do not spread to other part of your body, and they will be causing very little damage to your tissue. But when we are talking about malignant tumor, they means bad or harmful. They have neoplastic cell, right. There will be unlimited proliferation, grow very rapidly and invade and damage other tissue. There will be metastasis, and what do you mean by metastasis? Forms new tumors by displacement. They will migrate to other place, and NCERT is saying that most feared property of malignant cells or malignant tumors is this metastasis criteria, right. What do you mean by metastasis? Again I am explaining it. If this is your body, and here you got a tumor, a malignant tumor. Now few cells will be loosened up and will get mixed with your, will come into your blood circulation, and by following your blood circulatory pathway, it will establish another tumor into another part of your body. Within few days, it will establish another tumor into a third or into a different zone, and that condition is called as metastasis property of malignant tumor. Now if we talk about types of cancer, okay, so first is carcinoma. then it's sarcoma, then it's called lymphoma and leukemia. Okay, now what do you mean by carcinoma? Carcinoma means cancer of epithelial tissue, cancer of epithelial tissue like skin cancer. Okay, sarcoma, cancer of mesodermal tissue like bone cancer or cartilage cancer are normally called as sarcoma. Bone and cartilage both are mesodermally originated. Now lymphoma and leukemia, generally they are the blood cancer. Okay, lymphoma or leukemia, right? Leukemia, there is excessive, there is abnormal count of WBCs. That is the name of leukemia or blood cancer. Now in your blood, what's happening? The count of WBC will increase and those WBC will try to attack your own tissue. Okay. Now we'll talk about cancer detection and diagnosis. Okay. Now it's said that always early detection is necessary, as because if cancer is having multiple phases, first stage, second stage, third stage, right? Now generally you can hear in some movies or in some in TVs, that person is having third stage of cancer, doctor will be saying it's too late, we cannot save him or cannot save her. So if it's detected into the first stage, whenever inside your body a malignant mass of cells are developing, if it's detected then only, few kinds of cancer can be cured completely. Yes, NCERT is saying if it's detected into earlier stage, first stage, few, not all kind of cancer, but few kind of cancers can be cured, like breast cancer, right? Okay. So how to detect that? We can do blood examination and we have to check for increased WBC count, or there can be detection of tumor markers. Whenever there is abnormal cell or malignant tumor formation, they will start releasing some abnormal proteins. So those are called as markers. So if there is cancerous cell present into your body or developing into your body, in your bloodstream, in your body fluid, if I search for them, I will be getting those abnormal proteins or abnormal antigens. They are called as tumor marker or cancer marker. You have to search for these marker genes. Okay. Now, in case of leukemia, there will be increased WBC count, or if there is alkaline phosphate crossing a particular limit, a special chemical, then there is a chance that you may be diagnosed with bone cancer. If there is bone cancer, there will be increased concentration of alkaline phosphate into your plasma, into your blood. Okay. Sometime biopsy or histopathological studies are also done. Now, what you have to do if you are performing biopsy? The suspected tissue cut into thin sections and then stained and then examined under microscope. Okay. So if a doctor is suspecting that a particular tissue is having that particular cancer, suppose someone is having breast cancer, so with proper procedure, proper methodology, the tissue from breast will be collected, then they will be stained and they will be observed under microscope, and then the pathologist will give the conclusion either any abnormalities or markers are present into that or not. Okay. Now, if you want to detect cancer from internal organs, what you can do? You can perform simply radiography, CT scan, computed tomography and magnetic resonance imaging, MRI. So radiography, CT scan and MRI is done when you want to check for the cancer into internal organs. Okay. Now another method, antibodies against cancer specific antigens. So as I said that if a cell has converted into malignant or cancerous cell, then it will keep on releasing some abnormal proteins or abnormal glycoproteins into your blood. Now they can be detected by injecting some special cancer specific antibodies, as because this cancerous proteins gonna bind with this cancer specific antibody. If they are binding with each other like lock and key, if antibodies are binding with antigens, as because these antibodies are nothing but cancer specific antibodies, then this is a confirmatory test that that person is carrying cancer or abnormal carcinogenic cells, abnormal malignant tumor inside the body. Okay. Now few other techniques of molecular biology can be used. Here we can take help of biology. PCR can be performed, polymerase chain reaction, then autoradiography, PCR and autoradiography, some techniques of molecular biology can be performed to check for abnormal proteins or abnormal cells or abnormal genes. Generally in case of cancer, gene will be mutated. So if you want to check for that mutants, that mutated portion of your genetic makeup of your genome, you have to perform PCR, then gel electrophoresis, then southern blotting, and ultimately by the process with the help of autoradiography it can be confirmed is there any mutation present into my genome or not. Okay. Now how to proceed for treatment. Okay. Surgery can be done, like in case of breast cancer, the infected or the breast which is containing the malignant cells, with surgical method that can be eliminated. Radiotherapy can be given, okay, with the help of some ionizing ray, right? We can eradicate some cancerous cells. Chemotherapy also can be given with the help of some special chemicals. Administration of some special chemicals will be given into your body, and with blood circulation or with tissue space, inside the tissue space they will be helping us to eradicate or kill our cancerous cells. But remember, if you are performing chemotherapy with cancerous cell or malignant cells, sometimes they can kill normal cells of our body as well. But nowadays, while they are performing radiotherapy and chemotherapy, modernized medical has given us equipments to perform this technique in a more specific way that can only be targeting the cancerous cells or malignant tumors, and there will be no side effect of that. Okay. Now in some cases immunotherapy can be given. Now what is immunotherapy? Some biological response modifiers like alpha interferons are administered into our body. Now how they will be doing their function? Alpha interferons or biological response modifier. You know that in normal conditions, cancerous cell will be ignoring your normal immune system. Your immune system will try to kill your cancerous cell, but your cancerous cells are so much powerful, they will be blocking your immune cell attack. Now this alpha interference will somehow will make those cancerous cell timid or silence, and your immunity, your antibodies can kill those malignant tumors or cancerous cells. So basically, by administering alpha interferons, you are making cancerous cell weak, and this is advantage for our own immune system, and now our immune cells will be eradicating or killing or eliminating those malignant cells. So that's why we are naming these alpha interferons as biological response modifier. They can modify, they can make the cancerous cell much weaker. Okay. Now we are moving forward to the next topic, that is drug and alcohol abuse. If you have any question from cancer, you can write down your query into the comment section. Naga Raja KG is making some interesting emojis into the comment section. Recently, I was watching a meme in a class. The class teacher was saying everybody here will be talking only in English. Now there was a little guy inside the class, little boy, and I think his name was Nagaraja. Okay. Now, as because he was in peer pressure, everybody is talking in English, but now how he'll be pronouncing, how he'll be telling his name as Nagaraja. So he said, her teacher asked, his teacher asked him that, tell me what's your name. So he answered, my name is Snake King. Now that teacher got amused, Snake King, what kind of name is that? Now his friend murmured from like from the similar bench that, madam, his name is not Snake King, his name is Nagaraja. He's translating it into English. Okay. So drugs which are commonly abused. So these drugs are named as opioids, cannabinoids, and coca alkaloids. So we'll be focusing on these three kinds of drugs: opioids, cannabinoids, and coca alkaloids. What is the source of these particular drugs? How they can affect our physiology, okay, and how they are taken. Now, mostly these drugs or these narcotics drugs are obtained from flowering plants and some from fungi. You need to remember this. Most of them are obtained either from flowering plants or from fungi. Like if you talk about opioids, okay, so they are extracted from latex of poppy plants. Now, these are poppy plants. Now, in some part of our country, poppy seeds are used as an ingredient of our cooking material or in our kitchen sometime for many dishes, right? But cultivating these poppy plants without the consent of government is illegal, and most of the time those who are cultivating, those who are culturing these poppy plants, they are actually obtaining the latex, then drying it, and from that they are getting different kinds of opioid drugs. Okay, what is scientific name of poppy plant? You need to remember, that is Papavir somniferum. Papavir somniferum. And the prime opioid is consumed that is called as morphine. Yes, and in our NCERT, this is this chemical structure of morphine is also given, and you need to remember, you need to check once the diagram of opium or the poppy plant. Directly you can get these diagrams or get this structure. You can be asked uh, which of the following drug is that? Is it opioid, coca alkaloid, cannabinoid, or none of them? This kind of question can be asked. Okay. Now another modified version of morphine, that is called as heroin, and it got a common name called as smack. Okay. Now how heroin is made from morphine? It's actually diacetyl morphine. The process of acetylation of morphine, by the process of acetylation of morphine, heroin is prepared. So that's why then you can check the chemical name diacetyl morphine. It's white in color, odorless, bitter in taste, and crystalline compounds. Okay. So this question is often asked. Smack is a common name of which of the following drugs? You need to remember, smack is common name of heroin, and heroin is derivative of what? So heroin is derivative of morphine. Okay. Now how it's taken? It's taken by snorting or directly by injecting. Here you can check how snorting is done. Okay. And how it will be affecting your body. It act as a depressant, means it slows down your body activity, right? That's why it's called as depressant. When you are in depression, you are silent, you are very much quiet. A similar kind of condition uh, is like you will be feeling that your body is getting slow down, your heart rate, you are unable to think anything, the similar kind of condition. So the mechanism of action we have to study to understand how it's acting as a depression. Basically the morphine molecules or the opioid molecules, they binds to specific opioid receptor of our central nervous system and gastrointestinal tract. Okay. So the receptor for opioid, they are present in our central nervous system and GI tract. This also very much important line question can be asked. Okay. So whenever you are reading this topic or you are studying or understanding this topic, there is more to write down the things in pen and with pen and paper, and to remember for long term. What kind of question can be asked? Already I said that from which plant opioid is extracted. Okay. Smack is common name of what? Next question can be asked as for which of the following drug is a depressant? So these opioids are depressants. Now next thing you need to remember, important thing, the receptor, opioid receptors are present in to which part of our body, which organ? They are present on our CNS and our GI tract. Okay. They are also effective as sedative and painkiller. So that's why many of the, as a painkiller, sometimes morphine is given. Okay. Useful who have undergone surgery. Yes, after surgery sometime morphine was given as painkiller. It depends upon the dose. Okay, for a drug, a certain dose can be used as a medication, and over the dose or excessive dose will act as narcotics, will affect you badly. Next, we'll talk about cannabinoids. Now, these cannabinoids are obtained from where? From the inflorescence, means flower and flower related part. Inflorescence of which plant? Cannabis sativa. Yes. Now, which parts are used generally? Flower top or leaves or resins are used in various combinations to produce marijuana, hashish, charas and ganja. So marijuana, hashish, charas and ganja, all of them are basically cannabinoids. And how they are produced? To produce like hashish, you need resin of cannabis sativa plant, right? Okay. So it's, that's why it's said here, it's written here, flower top, leaves, resins are used in different proportion to form sometime marijuana, sometime if you change the proportion, like if there is more proportion of leaves and less proportion of flower top, it will become ganja, right? If you put some buds of flower or flower top along with the leaves, it will become marijuana. So different proportion you can from different cannabinoid drug. Why they are called as cannabinoid drug? Because they are obtained from cannabis sativa plant. Okay, how they are taken? They are taken by inhalation or by oral ingestion. Either you can inhale the smoke, right, and they affects the cardiovascular system. And mode of action you to remember, they interact with the cannabinoid receptor. Now question can be asked, where inside our body cannabinoid receptors are present? Cannabinoid receptors are present in our brain, and sometimes it's also used by some sports person because they have cardiovascular activity. So if a sports person is taking by following a particular limit, then it will make him or her tension free. The stage, there is a term called stage fear. Okay, Isha Pandey, you can get the notes of these classes into our WhatsApp channel, and you check this channel, the link given just here with this video in the comment section. The first comment you can check, and their WhatsApp channel link is given. You have to click that, and that will redirect you to our WhatsApp channel. After every lecture, in the next day, these notes will be uploaded. Okay, so what I was telling, that few sports person, they will be taking these cannabinoid drugs is because there is a term called stage fear. Okay, so if you are a sportsman and you have taken this cannabinoid in a particular limit, so that will eliminate your stage fear and that will enhance your performance. Okay. Now next drug is coca alkaloid or cocaine. Okay. Now from where we can obtain coca alkaloids? From coca plant, right? Now what is the scientific name of coca plant? Erythroxylum coca, and it's a native plant of South America. Commonly coca alkaloids are called as coke or crack, and usually they are snorted and they interact with the transport of neurotransmitter dopamine. See, dopamine is one of the happiness hormone or feel good hormone, right? If there is more secretion of dopamine, then your nervous system will get a stimulus. Yes, I can do the work, I have to do the work. So sometimes sports person also taking this coke or crack prior to their game, so that dopamine is released, they can retain more concentration of dopamine, right? And if there is more dopamine is acting with your neurons, is giving signal to your neurons, then they will feel energetic and it will give a boost towards their performance. But remember, the use of cannabinoids or coca alkaloids for sports person or just prior to game, it's banned. You might have heard about dope test, right? Okay. Now we'll talk about mode of action of cocaine or coca alkaloids. They stimulate action of our central nervous system, produces sense of euphoria. Yes, as because there is secretion of dopamine, there will be a feeling or sense of joy will be there, feeling of energetic, you will feel energetic suddenly. So that's the sense of euphoria, and that will be increasing energy. But excessive dose of cocaine can cause hallucination. Hallucination means if you take excessive dose of cocaine, whatever you are imagining, you can observe the things in front of your eyes, but this actually not happening. It's your imagination that's reflected in front of you, right? So that's called hallucination. Okay. So plants with some other plants with hallucinogenic properties. See, this is Atropa belladonna and this is Datura. Okay. So from Datura, the seeds are containing daturine. They can also act as hallucinogenic agents. Okay. Now, few other drugs called as barbiturates, amphetamines and benzodiazepines are normally used as medicines to help patients to cope with mental illness like depression and insomnia, because they also have a kind of depressant effect on central nervous system and cardiovascular system. So if someone is going through the phase of depression and insomnia, insomnia means where you cannot sleep, okay, because if you are too much tensed, then as a tranquilizer, barbiturates or amphetamines injections are given. So that will relax your muscle, relax your brain, and you will be falling asleep. Yes, Miss Sukriti, how you are doing? Okay. So these are often abused, even these barbiturates, amphetamines, which are generally for medication purpose used as tranquilizer. Okay, they are often abused. They are taken in high dose, and smoking also paves the way to hard drugs. So if you are a teenager, if you have entered into your age of adolescence, right, then few of your friends will be telling you that smoking is a kind of heroic job, right? If you are depressed, if you are broken, sometime if you are tense, they will be suggesting you start taking like a cigarette or something like that. Then suddenly due to the peer pressure, you will start thinking, yes, if I smoke, so that will make me look like a man, or that will boost my energy, and gradually for one day you will be taking, for the next day you will be smoking, for the third day you will be smoking, but for the first time when you are taking all these things, you will feel very bad. A guilty kind of feelings will be there, but due to your peer pressure, actually they are your bad friends. They will be influencing you very badly to take those cigarettes and all. Then gradually that will pave your pathway towards alcohol, right? And from alcohol that will lead towards hard doses of drugs like heroines and charas and all these things. So that's why these things have a temporary good effect, but long term they are gonna destroy your life. Okay. So tobacco has been used by human for more than 400 years, and tobacco can be smoked, can be chewed, or can be snuffed. Okay. Tobacco contains large number of chemical including nicotine and other alkaloids. Okay. Now, what is the function of nicotine here? Okay, the addiction you are getting, that is due to the presence of nicotine. Nicotine stimulates adrenal gland to release adrenaline and noradrenaline. So that's called adrenaline rush. When you are smoking, that will stimulate your adrenal gland to release more adrenaline, and that will give you a temporary or sudden energy, right? And the effect rise in BP and increase heart rate, and that's why you feel energetic. But cancer of lungs, urinary bladder, throat and oral cavity, cancer of oral cavity can be caused if you are chewing. Smoking even can also cause bronchitis, emphysema, the reduced surface of your lung. Okay, damaged alveoli will be there, coronary heart disease and gastric ulcer due to the presence of those aromatic components into that smoke. Okay, smoking increases carbon monoxide content of blood, and you know that if there is more than 1 to 2% carbon monoxide is produced inside your body, then they are going to react with your hemoglobin and they will convert your hemoglobin from oxyhemoglobin to carboxyhemoglobin, and then you may die. Okay, so that's how consuming nicotine by any form, the form of cigarette or gutka, is very bad for everyone. Okay, now once you have started taking the nicotine, that will give you a temporary adrenaline rush, and if you keep on continuing, then that adrenaline rush will give you some dopamine, right? And once you have stopped, then your brain will tell you something is missing from your body, and that will make an urge inside you to buy more and to smoke more. So that's how it's harming you. So what will happen then? Regular use of alcohol can cause damage of nervous system and liver cirrhosis or fatty liver, because liver is the detoxifier of your body, and if you are consuming alcohol, then what happens basically? In the beginning when you started, the alcohol will give inhibitory signal to your cerebellum, right? It will hyperpolarize your neuron, you will feel dizzy, and that dizziness may give you a short term or temporary charm. Now gradually if you start taking, so again there will be secretion of dopamine. If you are doing one thing again and again and again, so your body will start secretion of dopamine, and that will be giving you kind of adrenaline rush or feel a good feeling, and when you are not getting that, you will feel uh, like irritation will be there. Okay, but once you are addicted, that will be harming your organs. Specifically for alcohol, that is broken or detoxified, okay, by your livers, and if you are consuming too much of alcohol, your liver is working very hard for you and converting thus alcohol molecules into lactic acid, then glucose, then fatty acid, right? Then there will be deposition of fat, there will be damage of liver called as liver cirrhosis, and deposition of fats will cause this fatty liver, and when there is fat deposition or fat accumulation inside your hepatocyte cells, the cells of liver, they cannot perform the detoxification task. So gradually the liver will fail, and if liver is gone, gradually your kidney will also fail very soon and you will die. Now if you are trying to avoid these things, once you are addicted, now you are trying to withdraw yourself from the addiction. Now primarily what kind of symptoms can be felt by you? That is called withdrawal symptoms, right? So they are nausea, feeling of vomiting, mood fluctuation will be there, right? You are not getting that adrenaline rush, your dopamine secretion is not happening, that is not giving you a good feeling. There will be aches and pain, headaches will be there, stomachache will be there. You can face hallucinogenic effect. You may feel depression. There will be shakiness in your hands, right? There will be loss of sleep or insomnia, or sudden sweating can be felt by that person, but it's only for few days if you are feeling so, and if you really want to withdraw yourself from abusing drugs and alcohols or smoking, then you need to consult a doctor or counselor, or you can take help of your best friend or your parents or the person who loves you the most. So you have to divert your mindset, and that will gradually be helping you to recover and to successful withdrawal from this drug abuse. Okay, so we are done with the chapter human health and disease. Now if you have any doubt from any topic and you want to recap uh, the most difficult topics from this chapter, you can write down in the comment section. I will be explaining that again, or you can ask some question, then we will be moving forward to the next chapter. So again within 10 minutes we will start the new chapter. In the meantime, you can comment in the comment box that any topic from the chapter you want to understand in a different manner, or any tricky topic to you, then I'll be happy to explain it again. Or if you have any question, that also you can post into the comment part. Okay, so Nirika, we are talking about the side effect of the use of anabolic steroids in females and males. I guess this was not mentioned. Yes, that was not mentioned. Yeah, because that is means I haven't found any PYQs from this particular topic. That's why I've omitted that. Okay. So obviously, if you are taking anabolic steroid, uh, they are derivative of testosterone, right? Anabolic steroid are derivative of testosterone. So those who are doing sports activity or bodybuilders, right, for muscular growth and for their endurance, sometimes they will be taking anabolic steroids, but obviously anabolic steroids are very much bad for liver and kidney. Okay, so we'll start another chapter that is evolution. Okay, so before starting this chapter evolution, so can you tell me what do you mean by evolution? What do you mean by evolution? What is evolution to you? What is evolution to you? What do you think when you hear this term evolution? What comes into your mind? Like few may say the Darwin uncle is coming into my mind. Okay, someone is saying, Niharika is saying history of life, history of life. Okay, someone may say that from monkey to human being, those sort of things will be coming to mind. Okay, so obviously evolution is history of life, how from a single cell how from a single cell the life has flourished and forming over millions of species. Yes, history of life or history of life forms, how this diversity was created into this earth, and obviously we have to observe these phenomena in a huge background called this our universe. Yes, so that's the evolution. It's about making the diversity, right, from one species to millions of species. Evolution, few people will be thinking that evolution is about making advanced species from the inferior one. So it's not about that. It's about making the diversity into this living world. Okay. So if you want to study evolution, we have to study the formation of this universe, formation of this earth, and then in a small screen we have to observe, we have to peep in, and there will be observing how the life form started its journey, okay, back to somehow 4 billion years ago, and nowadays we can observe the life forms are so much flourished. Okay, and and one more thing, one very interesting thing that few people will be saying that when you talk about Darwinism, uh, that we evolved from monkeys. So what do you think on that matter? So what Darwin wanted to tell us, the evolution of human being. So generally the laymen are saying that Darwin said that human, we are descendants of monkeys. So is it real or something else? What do you think about the evolution of human being? It's our ancestor like monkeys or chimpanzees, or directly we were created by the Almighty. What do you think so? What's your opinion on that matter? Are we descendant of monkeys? No one is saying anything. So you people really don't have any opinion. You don't really love your subject. No one is writing anything. Come on, guys, write something. I asked about your opinion about Darwinism, about evolution of human, about Darwin's theory of evolution of human, his aspect. What do you think? Are we descendant of monkeys, or the Darwinism or the theory is represented in a wrong manner to us? Okay, so you don't want to say anything. Okay, so let's start the chapter. So if we talk about the unit, we'll be talking about some important terminologies. If you are asked what is unit of evolution, you have to answer it's population. Now what do you mean by population? Group of some individuals belonging from the same species. So they are called as population. So whenever evolution is started, uh, then it will be choosing some specific individuals from a population of a particular species. So that's why we say that unit of evolution is population. The driving force of evolution is natural selection. It's always nature selecting the superior ones. Okay. Now here the superiority means the individuals who are successful, who are fit in terms of reproduction, right? So they are chosen as the superior varieties. Now unit of natural selection, when nature is selecting the superiority, nature will be choosing some individual with some variations among them, and that's why it said that raw material of natural selection or raw material for evolution is gonna be variation or the variants. Okay, and the source of variation is generally comes from mutation, modification in the genetic makeup. Okay, so you need to remember this information. Now we will be exploring different theories of evolution. Okay, so let's check those theories. Like first theory, this theory of special creation. Okay, now according to the theory of special creation, which was given by Father Sorge, according to this theory, all living organisms are created as such. The diversity was always the same. Earth is about 4,000 years old. So, Father Sorge said that one day Almighty God thought that the superpower, the Almighty thought that he will be creating some species on the lap of earth, and that day only, within 6 days, only within 6 days, he sent all these species from the heaven, okay, from the some celestial part of this universe. And on the 7th day, the Almighty was taking rest, and that's why we count a week as 7 days. So obviously this is a mythological story, this is a religious story. We don't have anything to believe here. Okay, the next theory we will be exploring about, there is theory of panspermia. Now what do you mean by panspermia? It was given by early Greek thinkers and philosophers, and according to panspermia theory, some cosmic spores from some celestial entities are delivered into different planets, okay, and Earth was one of the chosen planet, and those cosmic pores, so those celestial pores came into Earth and flourished as life. So that is called as the panspermia theory. Yes, this theory is not absolutely baseless. There is some scientific basis of this particular theory. Now, next we will talk about theory of spontaneous generation. Now, spontaneous generation theory, that's a baseless theory. According to some old people, some laymen, that decaying and rotting matters like straw or mud is responsible for formation of life on earth. Okay. What they used to observe, if you keep a piece of meat, okay, in unwrapped condition, within few days that will start decaying, and the maggots of flies are hovering inside that. So they were thinking it's the decaying process of the meat which is responsible for formation of these maggots of this fly. So they had given their spontaneous generation theory, but obviously this was disproved by theory of biogenesis. What do you mean by theory of biogenesis? Means all living organisms are produced by their ancestors. Okay, so there is always relationship between ancestors and descendants. So it was given by Louis Pasteur, and Louis Pasteur, by some experiments, he disproved the spontaneous generation theory, and according to this theory, life comes from the pre-existing life, right? And after that we need to talk about modern theory, or the theory of abiogenesis, or the theory of chemical evolution, and that was given by these two scientists, Sir Oparin and Halden. So Oparin and Halden, they had given this theory of abiogenesis, or you can call it as protobiogenesis, or the theory of chemical evolution. So according to them, according to their theory, in the beginning of earth there are some inorganic constituent already present, right? Now under the influence of high temperature, volcanic storms and highly reducing atmosphere, yes, there was no oxygen, right, there was only hydrogen. So under these three conditions, there was formation of organic molecules from inorganic constituents. So this particular phase of creation from inorganic to organic, that is called as chemical evolution, and later on from organic molecules, combination of organic molecules given rise or stepped in life on earth. So from organic molecule to life formation, that is called as biological evolution. So for this theory, it is having two phases. First phase is called as theory of chemical evolution, second phase is called as theory of biological evolution. Chemical evolution means formation of organic molecules from inorganic molecules. Biological evolution means formation of life from organic molecules. Okay. So first self-replicating metabolic capsules were formed, right? Now while these organic molecules were forming the life, they were forming some self-replicating metabolic capsules, and you may call them either protobionts or eubionts. Okay, sometimes we call them quasarbates and macrospheres. Okay, so those things are not given into our NCERT, but you need to remember that how from non-living material or from some non-cellular components first cells came into existence. Okay, so those balls, those spheres are called as protocells, and they are this explained as first self-replicating metabolic capsule. Now let's check in detail how biogenesis or protobiogenesis took place. Okay, so for that we have to go back from here near about 13.8 billion years ago. Okay, now what happened 13.8 billion years ago? There was an enormously big blast happened in this space, in this entity, and that is called as Big Bang, right? And the theory of Big Bang was given by Lemaitre. And gradually after the Big Bang, the time and space of this universe was created, and this universe started expanding with time, right? And gradually the temperature was coming down, and as soon as the temperature was coming down, there was condensation of these gases, the primitive gases like hydrogen and helium, and due to the gravitation force, this condensation took place between hydrogen and helium gas and produced galaxies, and Milky Way is one of them. Okay, now inside that Milky Way galaxy there was a medium sized star called Sun, and from that star there was formation of this planetary system, we call it solar system. Okay, now inside the solar system there is a planet called Earth. Now the position of Earth, the distance of Earth from Sun is very much crucial for formation of life in it. Okay, formation of atmosphere is very much crucial for formation of life in it. Formation of ozone layer is very much crucial for formation of life in it. So how it took place? So near about 4.5 billion years ago, BYA means billion years ago, if we are doing a time travel, then we won't be we won't be finding any kind of atmosphere. Okay, there was molten mass. The earth was releasing molten mass. Okay, inside earth the molten mass is called as magma, and when they were coming out, we will be calling them as lava. Okay, so volcanic eruptions were going on, and they releases some water vapor, ammonia, carbon dioxide and methane. Okay, and gradually they were heated by ultraviolet rays which were coming from sun or the cosmos, and gradually by the influence of this ultraviolet started splitting water molecules into H2 plus O2, and this oxygen reacted with this ammonia and formed NO plus water vapor. This oxygen reacted with methane CH4 and formed carbon dioxide and water vapor, and the remaining oxygen molecules were binding with oxygen atoms under that volcanic storm, under that ultraviolet rays, and ozone gas was formed, right? Now gradually that ozone was forming a covering surrounding the early atmosphere. Okay, and that was very much crucial for formation of life, because without ozone there is nobody to block this ultraviolet rays, and if ultraviolet ray keep on continuing and to keep on continuing and stacking again and again the earth's surface, then formation of life, biogenesis was near to impossible. Okay, I am repeating. So 13.8 billion years ago there was unimaginable, enormously large blast, okay, from a single point, right, and that phenomena is called as Big Bang, and that created our universe, right. Now gradually temperature was coming down as because the universe started expanding, right, and due to the gravitation force the primitive gases like hydrogen and helium, they started coming closer and formed a cloud of helium and hydrogen, and those clouds were called as galaxies. Now one of those galaxies were our own Milky Way, right. Inside the Milky Way there was formation of sun and gradually the planetary system, the solar system, and inside the solar system Earth was a planet which was maintaining a perfect distance from sun, uh, which has that probability, the potentiality to produce form life inside it. Okay. Now let's do a time travel to that ancient Earth. We have to go back 4.5 billion years ago, right. So what you can observe, there is no atmosphere, the morning, the daytime is too much hot and the night time is too much cold. If there is no atmosphere, daytime will be very much hotter and night time will be very much cooler. Okay. So what is happening, there was molten mass was coming out from the solidified earth crust, right, and there was volcanic eruption and volcanic storms were happening, and the earth's surface was heated by random ultraviolet rays from the cosmos, means which was coming from different celestial bodies. Okay, and the initial gases, these were coming out from those molten masses are like water vapor, methane, ammonia, carbon dioxide, and gradually under the influence of ultraviolet ray they started reacting with each other, right, and that reaction accidentally or fortunately, we can say, not accidentally, fortunately, produced ozone gas when oxygen molecules will be reacted with oxygen atoms. There was formation of ozone. Now as because ozone is little bit like lighter, it tried to escape the earth's gravitation, but somehow it was trapped, right, and it formed a canopy, a covering surrounding the earth. Gradually the atmosphere was forming, and this canopy, this covering of ozone is very much important. Without this ozone layer, there was nobody to check to block the ultraviolet this UV hits, right. Okay. So gradually temperature was cooling and there was rainfall, okay, and there was formation of a primitive ocean, right, and you can call this primitive ocean as primordial soup. Why that primitive ocean is called as primordial soup is because the temperature of atmosphere was nearby 800 degrees Celsius and literally the ocean was boiling. So that's why better to call it as primordial soup. Okay. Now inside that ocean there was formation of some organic compounds from some inorganic constituents. Okay. Now what are those organic compounds? They are RNA, proteins, lipids, carbohydrates. Like now first sign of life was observed. Obviously, we haven't observed that, but according to the theory, first sign of life was observed 4 billion years ago by the cosmos, obviously. And what do you mean by first sign of life? Some biomolecules. What are they? They were RNA, proteins, sugars and nitrogenous bases. Remember, there is no DNA. Okay. And from them, they, they started coming closer to each other, right? And there was formation of first non-cellular life near about 3 billion years ago. Okay, now what do you mean by non-cellular life? There is a formation of cell-like thing, a sphere kind of structure, without any nucleus, without proper genetic material, without cytoplasm, without cell membrane, without cell wall, nothing was there, but a sphere that has the capability to replicate. So, that self-replicating non-cellular entity, we used to call it as protocell, right, or protobiont. From the protocell, eubiont came into existence, and nowadays we call it as macrosphere, right. And gradually, upon developmental phases, they had given rise first cell near about 2 billion years ago, and the very first cell which appeared into, which were thriving into that hot soup of the ocean, they were chemoheterotrophs. Remember, the first cells were not autotrophs, they were chemoheterotrophs. So, the chronology is like that: 4.5 billion years ago, Earth came into existence. 4 billion years ago, first sign of life, means biomolecules, came into existence. 3 billion years ago, first cell-like thing, a protocell, came into existence, and 2 billion years ago, first cell, that is chemoheterotrophs, came into existence inside that ocean. Okay. Now, from chemoheterotrophs were evolved. Now, what was the reason behind this evolution? Chemoheterotroph used to feed upon the biomolecules present into that ocean. Now, if you keep on feeding, then one day what will happen? There will be scarcity of the biomolecules. You will eat all the molecules from ocean, and the ocean is empty now. You have nothing to eat. Now, there is a need that you have to produce your own energy. You have to prepare your own food inside your body by utilizing the natural molecules, by utilizing some inorganic molecules present into the nature. So, that urge given rise some chemoautotrophs by utilizing hydrogen sulfide, by utilizing methane. They used to prepare their food, and gradually chemoautotrophs started evolving into photoautotrophs. Chemoautotrophs started evolving into photoautotrophs. Who are photoautotrophs? They used to harvest sunlight to produce energy, and there were two kinds of photoautotrophs were there. First one, anoxygenic photoautotroph, they used to release sulfur, okay, and the second type, oxygenic photoautotroph, they used to release oxygen. And from these oxygenic photoautotrophs, gradually they used to release oxygen. The oxygen were diffusing out from sea water for ocean water into the atmosphere, and gradually the reducing atmosphere started converting into oxidizing one, because oxygen started accumulating into the air, okay, and from there only life started its journey. The journey of life was accelerated, okay. So, this is all about that protobiogenesis, right, how life came into existence in our earth. Now, to support the theory given by the Oparin and Haldane, few scientists were working in the background, and according to Urey and Miller's experiment, it was proved that life came into existence in the earth from inorganic components. So, how they did this? They performed a closed flask experiment. They performed a closed flask experiment, and they tried to imitate the ancient earth. So, what was the condition of ancient earth? There was methane, hydrogen and ammonia. This kind of gases were present into the atmosphere. The atmosphere was highly reducing, right? UV rays were stucking or hitting the earth's surface. High temperature was there, near about 800 degrees Celsius. Volcanic eruption, volcanic storms and heavy rain was happening. So, this was the condition, the harsh condition of the primitive earth, where the atmosphere was reducing. The similar condition they have to imitate into laboratory. So, for that they have taken this setup. This is called closed flask setup, and inside that flask they have kept the same gases present into ancient earth's atmosphere, that is methane, hydrogen and ammonia, and they followed the ratio 2:2:1. Methane is to hydrogen is to ammonia. They have kept water vapor in that closed system. Temperature they used to maintain, that is 800 degrees Celsius, and they cannot get ultraviolet ray bombardment or volcanic storm. So, to supplement that, they had given electric discharge of 17,000 volt. Okay. Now, what will happen after some time? They observed gradually the methane, then ammonia, then water vapor and hydrogen started reacting with each other and started formation of organic components as amino acids, and they collected those amino acids and they found that glycine, alanine and aspartic acid, these three amino acids, you need to remember, glycine, alanine and aspartic acid were formed inside that closed flask. Okay. So, that was a victory of their research, and that supported the theory of abiogenesis or the chemical evolution theory given by Oparin and Haldane a lot. Okay. Now, we will close that chapter and we will jump with, we will try to explore with another legend of evolutionary biology, and that is Charles Darwin. So, let us talk about Charles Darwin. Now, he was very much keen, he wanted to know how the diversity is created, right. So, for that he went for a voyage, a sea voyage with the ship given by the British government, and name of the ship was HMS Beagle, Her Majesty's ship. Okay. And with that he visited different parts of the world and existing living forms, and what he concluded, existing living forms share similarities to varying degrees not only among themselves but also with life forms that existed millions of years ago. He got multiple fossilized animals, fossilized organism, and he found that the existing, the alive organisms, we have similarities and dissimilarities among us. Similarly, we have similarities with those fossilized organism as well. So, that is giving a clue towards that fact, we are appeared from a similar kind of ancestor. Like the journey of life was like this. There was a life form called A, Gradually there was evolution, formation of two new form of life called A1 and A2. From A1 there was formation of B and B1. From A2 there was formation of A3, C and B2. So in this manner life keep on moving, and in the course of time few got extincted, but though they got extinction, they had to face extinction, but they share some similarities with modern day organisms, and that's what Darwin sir wanted to tell, that evolution means is the journey of life from a single species to millions of species, how the living world was diversified. Okay. So there has been gradual evolution of life form, means according to Darwin, evolution is a very slow process. There will be gradual accumulation of variation generation by generation, and suddenly one day there will be formation of a new species. Okay. So while you are studying Darwinism, you have to counter with these words: high rate of reproduction is seen among all the animal species. Okay. And obviously if you are performing sexual reproduction, there will be chance of formation of variation due to the due to recombination in between the chromosomes, right. So that will result formation of variants, and variants are also called as descent with modification. Variants are also called as descents with modification. Okay. Now under certain circumstance, if your species is abolished or extincted, maybe the variants will manage to survive due to their changed nature, right. So that is the importance of descents with modification, that is the importance of variants. Okay. But the resources are limited, the landforms or the water bodies are limited, but organisms are expanding or increasing their number in exponential way, right. So what will happen after some day, there will be competition among them. There will be intraspecific competition. Man and a tiger will fight with a tiger, man will fight with another man, a lion will fight with another lion, that is called intraspecific competition. Then there will be interspecific competition, a tiger will fight with a lion, a man will fight with a chimpanzee, that is called interspecific competition. And obviously the natural calamities is the natural competition for us. Okay. Disease, epidemics, pandemics are also competing with the progression of journey of life, and that competition is called as struggle for existence. Everybody wants to win the race, so everybody is competing, everybody is giving their effort. So that's why everyone is in struggle, everybody is struggling to survive, that is called struggle for existence. But see what will happen, the survival of the fittest will be there, but the survival of the fittest will be there if you are managed to reproduce your progeny under adverse circumstance, then your species will survive. If you are unable to reproduce under the modified, under the changed environment, under the changed climate, then there will be extinction. So that is called survival of fittest, and this particular terminology, survival of fittest, that was adapted by Darwin and was actually given by Herbert Spencer. Okay. Now if you want the explanation of this survival of the fittest, Now, if you think about the Jurassic period and Mesozoic era, who was the king of this planet? Is the dinosaurs. T-Rex, remember, very dangerous animals. They were reptiles. There was giant reptiles roaming here and there. Okay, they were killing all other animals. They were just ruling over the earth. But what happened? One day there was a asteroid attack, or there was seasonal change, ice age came, and as because reptiles, they are cold blooded animal, they were not able to manage their survivability into the ice age. So most of them died. Only the smaller ones survived, and from them there was emerge and from then only birds and mammals started emerging as the ruler of this planet, and nowadays, okay, mammals are ruling. So how the thorn has changed the ruler? It's the nature. Nature is choosing the fittest species. Is not the strongest physically. It's not the physical fitness, it's about the reproductive fitness. So that's what Darwin wanted to say. Okay, and that process, seamless process, is called as natural selection. That choosing reproductively fit organisms, okay, in the journey, in the race of this life, is called as natural selection. Okay. Now this similar kind of observation was also given by Sir Alfred Wallace. Whatever Darwin told us, Darwin observed. Specifically, if we check Darwin's research on Galapagos Island and all, the similar kind of research was carried out by Alfred Wallace on a different piece of land that is called as Malay Archipelago, that is nowadays that is present in Indonesia. Okay. So according to him, Sir Alfred Wallace, all the existing life forms share similarities and share common ancestors. However, these ancestors were present in different period of history on earth. The similar thing, same but different, but similar thing, but in a separate manner, in a different manner, was told by Sir Alfred Wallace. So if two pioneer scientists, they are giving their conclusion, and you are finding, as a learner, you are finding both of them are concluding the journey of life in a similar way, then there must be some truth. And that's why today in our chapter we are following Sir Darwin. We are talking about Sir Alfred Wallace, and that is the reason the scientific community of the whole world, they are behind Darwin, right, and Darwinism. Okay. Now we'll talk about evidences for evolution. Now we'll talk about evidences for evolution. Now, if you want to prove theoretically, that's fine, but you have to give some real proofs, so that the people who are questioning the method, the mechanism of evolution, right, you can answer them in a proper way. So that's why these evidences will be helping us, or these are the justification behind the theory of evolution. First, we have to check the paleontological evidence. Paleontology means study of fossils. Okay. Now, what do you mean by fossils? Rockified bodies of plants and animals under the rock, under the earth crust, due to the extreme temperature and pressure, they actually got rockified. Okay, but their body design is still intact, their impressions are still intact. they are very much beautiful. Okay. So different sediments of rocks will actually exhibit different life forms, and they existed in different geological time scale. Okay. Certain life forms are restricted to certain geological time span, and that denotes, let me explain into a different way. Suppose this is the piece of land, piece of earth crust, and these are the different layers. This is the first layer, this is the second layer, this is the third layer, this is the fourth layer. Now what you have to do? You have to excavate. You have started excavation, digging the earth. Now you will be finding different layers of rocks, with different component, right? And suppose you found that this layer of rock is having mostly the fossils of fishes. This layer of rock is having mostly the fossils of fishes are there. For this layer, mostly the fossils of amphibians are there. For this layer, mostly the fossils of reptiles are found. So you can, it's not very much tough to guess that, this layer of rock is telling us the story of history, that suppose some few millions of years ago in the earth was inhabited or the ruled by fishes. Okay, and after some time they got extincted, they died, and now it was time for amphibians. And after some time similar thing happened, all these amphibians, they died, and now it was time for reptilians. So if you keep on digging, the deepest layer will tell us about the primitive animals or primitive organism, and the superficial layer and the fossilized animals will tell us story about the recent animals. So that is called as geological time span or geological time scale. And with the help of some scientific mechanism, scientific study, you can actually measure the age of this layer of rock or this layer of rock or this layer of rock, and we call the methods as dating techniques, like radiocarbon dating techniques, radio other radio dating techniques, multiple radio dating techniques are there. Okay. So geological time scales are divided into this manner: eon, era, period, epoch, and age. And NCERT has asked the similar question: how the ages of fossils are calculated? Ages of fossils are calculated with the help of dating techniques. Like the most common dating technique, that is radiocarbon dating. Okay. Now, how radiocarbon dating is performed? Utilizing radiocarbon dating, how you can calculate the age of a fossil? See, while you are inhaling two kinds of carbon. One is C12, that is the normal carbon, and the radiocarbon, that is C14. Okay. Now this radiocarbon C14 will be exhibiting radioactivity, right? Now what we have to do, if you are finding a fossil, you have to check the ratio. You have to check the ratio of C14 and C12, and that ratio will help you to determine the real age of that particular fossil or that particular rock. Okay. Now next evidence, that is embryological support or embryological evidence. that was given by Ernst Haeckel. And according to him, ontology recapitulates phylogeny. That means ontogeny recapitulates phylogeny means descendants try to repeat the characteristics feature of the ancestors. Okay, if you translate into English, right? So ontogeny means descendants, recapitulate means try to recap, try to repeat, and phylogeny means ancestors. The ancestral characteristics features are still present into descendants, right? So that is the meaning of this particular sentence. So according to this theory, Ernst Haeckel tried to explain that some common embryogenic stages among vertebrates, like if you check, if you take embryo of shark, embryo of crocodile, embryo of penguin, embryo of cheetah, and embryo of human being, these are different vertebrates, right? Shark belongs to the group fishes, right? If you take one embryo from a fish, if you take one embryo of amphibia, if you take one embryo from reptile, one embryo from bird, and one embryo from mammal, then it's quite amazing to study the embryonic phases. All of them are posing gill slits. All of them are posing gill slits. Though we are not aquatic animal, we don't need gills for respiration, but our embryonic stage, even embryo of human will exhibit gill slit, but they are actually vestigial gill slit, non-functional gill slits. That means our embryo is trying to repeat the characteristics features which were present into our ancestors. Okay. So gill slits which are non-functional in human embryo, but the gills which are functional in fish. So obviously, if we study evolutionary history, the evolution is like that, from fish to amphibia, from amphibia to reptilia, then from reptilia to bird and mammal. So who is descendant here? We are descendant. Who is our ancestor here? Fish is our ancestor. So our embryo is trying to copy our ancestral feature inside that, and that's how Ernst Haeckel tried to prove that these embryonic evidences are basically giving us a denotation, a signal that we are this evolution took place in this earth, and the chronology was like this: fish, amphibia, reptile, birds and mammals. But another scientist called Carl Ernst von Baer, he tried to disapprove the theory given by Ernst Haeckel. So this kind of question can be asked. Embryological support, the theory was given by Ernst Haeckel, and who tried to disapprove it? Carl Ernst von Baer. So a famous statement given by Ernst Haeckel: ontogeny recapitulates phylogeny. What is the meaning of that? Descendants means a new organism, a modern organism will try to repeat some feature into the embryonic phase which were present into its ancestor. Okay. Now, why this Carl Ernst von Baer disapproved the theory? He said that's fine that you are getting embryonic common embryonic feature among different groups of vertebrates, but why the gills are present into embryo, vestigial non-functional gills, but while the baby is parturated after birth, why the baby is not pausing the gill slits? Okay, so that means there is no scientific basis of this theory, and by saying that, Carl Ernst disapproved the theory. Okay. Now the next thing we need to talk about: comparative anatomy and morphology. And when we talk about comparative anatomy, first we need to talk about homology and then we need to talk about analogy. So what do you mean by homology? What is difference between homology and analogy? Homology means similar origin and structure, but different function. Analogy means similar function but different origin and structure, right? So homology tells us about common ancestry. Homology tells us about common ancestry and divergent evolution. For example, if you try to differentiate between forelimb of different vertebrates, forelimb of man, cheetah, whale and bat, what you are gonna get? You are gonna get that forelimbs of all these different animals have similar bones, like they have humerus, all of them have humerus, all of them have radius and ulna, all of them have carpals, metacarpals and phalanges into their forelimbs. So that means somehow they were originated from some similar ancestor. Okay, and that is the meaning of divergent evolution. That is the meaning of divergent evolution. Similar ancestor, and from the similar ancestor, different groups spreaded or diverted into different direction of the world. That's why there is dissimilarities among them. But if you track down their ancestry, you will be finding that they are coming from the same place, from the similar kind of organism. So this is called as divergent evolution. Okay. Another example as given in NCERT, heart of vertebrates. Heart of vertebrates. How from 2-chambered to 3-chambered and 4-chambered heart was evolved. Now if you check the plant kingdom, then throne and tendril of bougainvillea plant and cucurbita. Throne and tendril of bougainvillea plant and cucurbita. Okay. So we are talking about similar origin and structure. So both of these structures, throne and tendril, they are modified stem, but they are performing different function, right? Similarly, for forelimb, we are utilizing our hand to write, to hold, to eat. Cheetah is utilizing the forelimb to run. Whale is utilizing the forelimbs or as a flipper to swim. Bats are utilizing forelimb as patagium to fly. So all of them are performing different function with the help of their forelimb, but they have basic similar structure as because their origin is same. Similarly, if we talk about analogous organ, now function is same but structure and origin is different. So that denotes convergent evolution or sharing of common habitat. For example, if we compare between insects, they also have wings, birds, they also have wings, right? And few reptiles, they also have wings, and few mammal like bat, they also have wings. So all different groups of animals you will be finding wings are present. So what is the function of wing? Help them to fly, right? So that is the similar function. But if you compare the wing of bird with wing of butterfly and wing of bat, that's completely different in structure from each other, right? So that denotes somehow at some point of time birds, insect, reptiles and bat, they came together, they were sharing similar geographical zone, and that is the reason nature compelled their body parts to get modified and form wing for flight. Okay. So examples are as given: wings of butterfly and birds, eyes of octopus and mammals, flippers of penguins and dolphins. Penguin is a bird, dolphin is a mammal. Okay, and the tuber, if you talk about plant kingdom, tuber of sweet potato and potato, both are performing same function, storage of food, but sweet potato is modified root, but potato is modified stem. That means different structure. Okay, now we will talk about some evidences from biochemistry. So, thing is that there will be similarities in proteins and genes performing given function among diverse organisms give clues to common ancestry. You know that genetic code is universal. You know that genetic code is universal. So, what can be a bigger proof of biochemistry? Means gene is coding for the triplet is coding for amino acid. That means gene is coding for a specific proteins. So, similar kind of proteins are produced, similar kind of amino acids. Okay, you will be needing only 20 amino acids, 20 proteinogenic amino acids are present in all the organisms. So, that is the biggest proof that somehow all the organisms are connected into history, and that gives us proof about diversification and evolution. So, till now from up to this topic, if you have any doubt, any question, you can write down your uh, curiosity or query or question into the comment box. Okay. Now, we will move forward to explore the next evidence of evolution, that is artificial selection and natural selection. So, why we need to compare between artificial selection and natural selection? We human, we are a creature created by this nature. We have very limited power, and nature is omnipotent, nature is God, nature is almighty, right? Now, if our effort can produce some new varieties, right, by the process of selective breeding, by the process of selective breeding, if we can produce new breed, so formation of a new species for nature, it's not a big deal, because nature has all the power. Nature is called as omnipotent, right? Like if I explain about artificial selection, you know, if you go to vegetable market, you can observe different kinds of vegetables, like one is broccoli, another is cabbage, another is cauliflower. Now, if you talk about these three vegetables, broccoli, cabbage, cauliflower, so apparently they look different. We think that they are different vegetables, but do you know that it's produced by human being from a process called selective breeding, okay, from wild cabbage. So, cabbage is nothing but a special selective breed of wild cabbage, cauliflower, a selective breed of wild cabbage, broccoli or kohlrabi, that is also selective breed of wild cabbage. Okay. All the different forms of dogs we can observe, like for in India we have street dog, we have German Shepherd, we have some Doberman, we have Rottweiler, we have some other like any other breeds of dogs if you talk about. Saint Bernard Shaw. So all these different breeds of dogs actually a resultant breed of wild dog or wild wolf kind of creature. Okay. So all these dogs came from the similar ancestor, and it's us human being who have chosen some particular characteristics feature inside them over another. So these different breeds of dogs are created by human being. These different breeds of vegetables created by human being. Different breeds of paddy, different breeds of wheat created by human being within short span of time. Now nature or the evolution is having billions of years, millions of years. So it's quite easy task for the almighty, for the omnipotent nature to create millions of species and the diversity. Okay. So when you talk about natural selection, you can give example of resistant varieties like antibiotic resistance and herbicide resistance. That is called anthropogenic action. It's very simple. To eradicate mosquitoes from your house, from your home, we buy this All Out or Good Night, the liquids, right? But if you keep on using the same brand, suppose you bought All Out, the liquid, and for 2-3 months you are utilizing the same thing, then after that what you will be observing, the mosquitoes are dancing, into your room, they are not bothering about the All Out or Good Night, unable to eradicate, unable to repel, as because they become resistance, right? So that is called as anthropogenic action. If you check the advertisement of Dettol, what you can hear, Dettol can eradicate, Dettol can clean 99.99% of germs from your home. But what about that 0.01% germ? Why they don't tell in their ad Dettol can eradicate 100% of germs from your home? That 0.1% become resistant by continuous or frequent use of Dettols or some antiseptic liquids. So those are creation of human being. Resistant, antibiotic resistant varieties, herbicide resistant varieties. Okay. We will talk about another anthropogenic selection, that is industrial melanism. Okay. So we need this basic knowledge that lichen is pollution indicator. Lichen, you know about the symbiotic relationship between fungi and algae, that is called lichen. So lichen is pollution indicator. Lichens will be growing upon the barks of the tree, barks of the old tree, wherever there is air is fresh, there is no pollution. Okay. Now the thing is like that, the case study before 1820, before the industrial revolution, there was more white moths, right? But after 1920, it's just after the industrialization or industrial revolution, naturalists found that there was more dark moth or melanized moth. But in rural areas, even after industrialization, there was quite low count of melanic moths but high count of white moths. Okay. Now one came and explained the mechanism, this is the mutation. Okay, as because before industrial revolution there was no pollution, and gradually when industrialization started, the pollution caused a mutation into their gene. That's why the white moths became black moths. But actually he was wrong. What happened here? We have to explain this mechanism by this anthropogenic action. What happened? See, before industrialization, uh, the tree, the barks of the tree, they had growth of lichen on them. So what should be the background of the forest? It's white, due to the presence of lichen on the bark of trees, right? Now, if the background is white, if the background is white and white moth is sitting on it, then you will be overlooking this, because that not creating a contrast. But similarly, on the white background, if a black moth is sitting, due to the contrast created, it will be easily visible by you, so as by the predator. So what happened before industrialization? The birds, actually the main prime predator for these moths, they used to observe these black moths due to their contrast, and they used to catch and eat them. So whenever you are observing in the nature, you are observing only white moths, right? But the scenario changed just after the industrial melanism. Due to the carbon particles, all the lichens, due to the pollution, the lichens died and the bark of trees became black. Now the background is black. And on black background, both the moths are sitting. One moth, the white moth, sitting on black background. Now this will create a contrast. But if the black moth is sitting on black background, no one can visualize it. So the scenario is reversed now. The same bird now started to visualize the same bird, the same predator started to visualize the white moth now, and upon overeating, gradually the population of white moth became very low and there was only black moths. So when you will be observing after industrialization, then you will think the city is having only black moths. So basically, before industrial revolution, nature selected the white moths over black moths, but after the anthropogenic action of industrial revolution, nature is selecting black moths over the white moth, and that is the reason behind the population of white moth before industrial revolution and high population of black moth after industrial revolution. And that's why we say that evolution is a stochastic process. That's why we say that evolution is a stochastic, means it's a random process. You never know when the nature will be selecting which variety, right? If you observe from the angle of nature, so before industrialization, who was better, who was fit? White moth was fit. After industrial revolution, from the angle of nature, from the vision of nature, who was fit? Black moth was fit. Okay, and that charisma of nature is called as that evolution is stochastic process, a random process. Okay, so If you have any queries or question, you can write down in the comment section. So if you don't have anything to ask, then I'm ending the class for today. Again, I'll be meeting tomorrow with some new chapter and the remaining syllabus of our curriculum.